Good morning. I have a great pleasure to welcome you all to this EFSA workshop on stakeholder, discussing about selection of comparators in risk assessment GM plans. I have a special pleasure to welcome Member of the Parliament, Vice Chair of the Acting Committee, José Bové, as well as Dr. Sebastian Gu from the European Commission, and all those who contributed to the public consultation on the draft guidance on selection of comparators. I would also like to thank those who are joining us today via our live webcasting, and also our audience with members of academics and representatives of industry and environmental groups. I'm also informing you with great pleasure we have here a moderator, Dr. Helmut Kalbitsch. He has a very long experience assessing the GMOs, and he is the head of unit of land and biosafety in the Environmental Agency in Austria. Before we begin, I give apologies to our Director General, Catherine Sefalen, who couldn't join in person today, but has been kind enough to offer us a short video message which we now will hear.
Dear Vice Chair of the Agriculture Committee of the European Parliament, Cher José Bové, distinguished guests, ladies and gentlemen, thank you for taking part in this important workshop organized by EFSA with its GMO panel. Our topic today, GMOs, remain highly controversial. A recent Eurobarometer survey on life science and biotechnologies shows that a clear majority of Europeans, 61%, remains broadly opposed to GM food. Although we know that perceptions of GMO are related not only to science, the need for robust, high-quality scientific advice is essential to protect public health and the environment. This is EFSA's role, and therefore, our work on establishing guidance on the selection of comparators for the risk assessment of GM plants is crucial. In EFSA's draft guidance, we use a comparative approach, the so-called principle of substantial equivalence. This is the starting point for risk assessment of GMOs, not just in Europe, but globally. We know that it is subject to questions at times. Given the important role that EFSA plays as Europe's risk assessor, providing scientific advice to the legislator, it is critical that we can foster open debates on science, including controversial areas. This is why we organized today's event. EFSA places great importance on consulting with you, its stakeholders, on its scientific work. Through this open dialogue, all parties have benefited from listening to different scientific views. The feedback received is deliberated at length by our experts and incorporated into our opinions as appropriate. With this in mind, I would like to thank my colleagues and EFSA experts, but most importantly you, for contributing to and advancing our important work on GMO risk assessment. I wish you all an excellent workshop and look forward to hearing about the outcome of what I hope will be a very fruitful debate. Goodbye.
I would like to add some things about Catherine's work. In recent public consultation, we received 139 comments from 18 different organizations, encompassing broad and often contradictory views and opinions. I'm pleased to say that a lot of those who contributed to consultation are here today and can take a floor to discuss these issues with us. This forum is here to allow the further discussion of views and opinions and engage directly with EFSA GMO panel members and regular members on this very important topic. The outcomes of today's discussions will be considered by the GMO panel when finalizing their opinion on the selection of comparators for GM risk assessment. Today's workshop will be split in two sessions. In the first session, there will be a general discussion about the concept and substance equivalence, with a view to giving some context to the way in which EFSA carries out risk assessment and GM plans. And this session also had a unit of GMO, Parapergama, will join us and helping us. During this session, we also have speakers and presentations made by Dr. Sebastian Gu from the Commission, Dr. Helmut from the European Network of Scientists for Social and Environmental Responsibility, and Dr. Becker Kearns from the Organization for Economic Cooperation and Development. In the second part of the day, we will focus on EFSA's draft guidance itself, hearing from EFSA panel and working group, as well as those who contributed for public consultation, views and comments during this session. The session is divided in different sections, such as risk assessment of single events, fact events not from conventional crossing, and risk assessment without any comparators. We are certainly looking for a lively and constructive discussion on science in the hours to come. I now have the pleasure to give the floor to José Bové, Member of the Parliament, Vice Chair of the European Parliament Committee on Agriculture and Rural Development, who will open our meeting today. Please, the floor is yours.
Alors, je vais essayer d'enlever ça. Mesdames et messieurs, tout d'abord bonjour. Je voudrais tout d'abord commencer en remerciant Madame la directrice, Madame Jalin-Lenné, pour avoir organisé cette réunion qui, me semble-t-il, est la première du genre, et je crois que c'est tout à l'honneur de l'EFSA. Il a fallu attendre près de 20 ans pour qu'une agence de sécurité alimentaire accepte d'ouvrir le débat sur le concept d'équivalence en substance. 20 ans de controverses scientifiques, d'opacité, pour qu'aujourd'hui enfin l'EFSA permette la confrontation des points de vue sur ce point central des divergences. Malheureusement, seules les 2 premières heures de cette conférence permettront de confronter ces 2 opinions sur ce sujet. J'aurais souhaité, comme Mme Jalin-Lenné l'avait proposé lorsqu'elle m'a rencontré à Bruxelles le 17 novembre dernier, que cette journée soit consacrée entièrement à ce sujet. L'intitulé de cette conférence, « Sélection des comparateurs pour l'évaluation des plantes OGM », montre que le thème a profondément changé, je le regrette. Malgré cette évolution, j'estime dommageable, j'ai néanmoins tenu à ouvrir cet atelier, car pour autant, je suis convaincu que ce temps de dialogue ne nous permettra pas de clore le débat. Il s'agit d'un premier pas. D'autres suivront qui nous permettront collectivement de trouver un moyen d'évaluer réellement l'ensemble des risques toxicologiques, environnementaux, sociaux et économiques liés à l'utilisation des OGM dans l'agriculture et l'alimentation. Je me permets de revenir rapidement sur les conditions qui ont permis l'organisation de cette journée. Le 14 juillet 2010, j'ai rencontré à sa demande le commissaire Dalli, qui préparait sa proposition de renationalisation des autorisations des OGM. Lors de cette réunion, j'ai entre autres alerté M. Dalli sur les conflits d'intérêts qu'il y avait à la tête de l'EFSA. En particulier, je lui ai expliqué que Mme Banati, la présidente de cette agence, était également membre du conseil des directeurs d'ILSI Europe, qui se trouve être un des groupes de pression les plus puissants et les mieux organisés sur les questions agroalimentaires, et que ce double engagement était de nature à influencer les décisions que vous pouvez prendre, en particulier en ce qui concerne les choix scientifiques qui composent le panel OGM. L'EFSA, créée en 2002, et selon le règlement européen du 28 janvier 2002, chargée, je cite, d'être une source indépendante en matière de conseil, d'information et de communication sur les risques pour améliorer la confiance des consommateurs. À ce titre, elle est donc obligée de choisir des experts scientifiques indépendants, des entreprises ayant des intérêts financiers dans les décisions qui seront prises dans le cadre des opinions scientifiques. Cette indépendance est absolument nécessaire. Le 14 juillet, j'ai demandé à M. Dalli que les services de la commission Sanco prennent des mesures nécessaires pour mettre un terme à ce conflit d'intérêts. Cette requête n'a pas été suivie d'été. Il a fallu attendre le 28 septembre 2010, une conférence de presse organisée au Parlement européen et des articles publiés à sa suite, pour permettent de porter à la connaissance du public ce conflit d'intérêts. Lors du conseil d'administration de l'EFSA, qui s'est déroulé les 20 et 21 octobre 2010, Mme Banati a finalement pris la décision de démissionner de ses fonctions de l'ILSI. Cette réaction montre à l'évidence que ces deux engagements n'étaient pas compatibles. Retracer l'histoire de ces quelques mois est nécessaire pour deux raisons. Cela montre d'une part la manière dont l'industrie des biotechnologies est parvenue à influencer les décisions de l'EFSA, en plaçant dans ses instances décisionnelles des personnalités scientifiques qui représentent ses analyses et qui partagent ses objectifs. Cela permet également de souligner que malheureusement, sans la mobilisation de l'opinion publique, les décideurs politiques de l'Union européenne, que ce soit le Conseil, la Commission ou le Parlement, ne sont pas toujours en mesure de conserver une position claire et impartiale sur des questions qui préoccupent leurs concitoyens. Après cette brève introduction, permettez-moi d'aborder maintenant le sujet essentiel de cette journée, l'équivalence en substance qui est au centre d'une polémique scientifique depuis 1993. Les premières variétés transgéniques utilisables à grande échelle en agriculture sont apparues à la fin des années 80. Les entreprises qui les ont développées, vous les connaissez, Monsanto, Pioneer, Dupont, Bayer, BASF, avaient besoin de la mise en place d'une réglementation la moins contraignante possible pour commercialiser leurs nouvelles semences. Pour elles, il était impératif de réduire au maximum les tests toxicologiques et les études d'impact sur l'environnement. Leur objectif était de persuader les autorités publiques et les consommateurs que les OGM n'étaient pas intrinsèquement différents des plantes conventionnelles utilisées jusque-là. En 1988, l'OCDE a mis en place un groupe de travail dont l'objet était de parvenir à un accord pour une réglementation globale sur les autorisations de ces nouvelles technologies. À cet effet, le groupe des experts nationaux sur la sûreté en biotechnologie, le GEN, a été créé. Certaines des personnalités qui ont contribué à ce travail étaient ouvertement favorables aux biotechnologies et à leur mise en place rapide sur le marché. Dès cette époque, l'industrie des biotechnologies était très active et cherchait à influencer la définition du cadre réglementaire en gestation. Je me permettrai d'illustrer mon propos par un exemple simplement, celui de M. Willy De Greef, expert belge. En 1991, M. De Greef est président du groupe national d'experts dont les conclusions serviront de base à la mise en place du concept d'équivalence en substance. M. Willy De Greef a également été responsable des questions réglementaires pour deux entreprises importantes impliquées dans les biotechnologies. Dans les années 1980, il a travaillé pour Plant Genetic System, qui sera racheté en 2002 par Bayer Crop Science. Entre la fin des années 90 et 2003, il a occupé un poste similaire chez Syngenta Seeds. Le 1er juillet 2008, M. De Greef est devenu le secrétaire général d'EuropaBio, l'association européenne des bioindustries, dont la mission est de promouvoir en Europe une industrie biotechnologique dynamique et inventive selon leurs propos. Il occupera cette fonction jusqu'en avril 2010, date à laquelle il deviendra directeur exécutif de l'IBRS, l'International Biotechnology Regulatory Services. Cette proximité entre d'une part le monde de l'industrie et d'autre part celui de l'administration jette de sérieux doutes sur l'impartialité des études qui ont été produites à cette époque et qui ont profondément influencé la mise en place des règlements visant à encadrer la mise sur le marché des OGM. Parallèlement au travail mené au sein de l'OCDE, les entreprises ayant des intérêts dans le secteur des semences se sont engagées dans une campagne de lobbying intensif. En 1988, elles ont créé l'IFBC, International Food and Biotechnology Council, qui publiera un rapport en 1990 intitulé Assuring the Safety of Food Produced by Genetic Modification, Regulatory, Toxicology and Pharmacology. Ce travail servira de base à la mise en place de la législation des États-Unis. Dans son rapport, l'IFBC conclut qu'aucune nouvelle mesure réglementaire n'est nécessaire pour encadrer des aliments ou des ingrédients alimentaires produits à partir de sources ayant été génétiquement modifiées. Ces recommandations peu contraignantes ont été adoptés l'année suivante. L'IFBC recommande également, ouvrez les guillemets, que le principal moyen pour évaluer des produits génétiquement modifiés repose sur une comparaison de la composition des nouveaux produits avec celle de ses équivalents traditionnels en ce qui concerne les niveaux de leurs éléments constitutifs. L'idée est énoncée. Un an plus tard, l'OCDE valide officiellement cette proposition et la baptise du nom d'équivalence en substance. Dans un rapport intitulé « Évaluation de la sécurité des denrées alimentaires issues de la biotechnologie moderne ». Nous sommes en 1991. Les auteurs de ce nouveau document considèrent, ouvrez les guillemets, le concept d'équivalence en substance et la concrétisation du principe selon lequel les organismes existants utilisés comme aliments ou comme source d'aliments peuvent servir de base pour la comparaison sur le plan de l'innocuité de la consommation par l'homme avec un aliment ou un constituant alimentaire modifié ou nouveau. En l'occurrence, l'idée d'équivalence substantielle ne repose pas sur une démonstration scientifique irréfutable, comme celle qui permet d'établir la loi de la gravité universelle, même si les scientifiques pontificaux de l'époque ne la reconnaissaient pas, mais sur la concrétisation d'un principe, c'est énoncé dans les documents officiels. Pour éclairer le propos, le Centre national des ressources textuelles et lexicales donne la définition suivante du principe. Un principe est une proposition posée au début d'une déduction, ne se déduisant elle-même d'aucune autre dans le système considéré, et par suite mise jusqu'à nouvel ordre en dehors de toute discussion. C'est pour ça que le débat d'aujourd'hui est effectivement une nouveauté. Dès le début aux États-Unis, des scientifiques au sein de la Food and Drug Administration ont exprimé des doutes sur le fait que l'équivalence en substance puisse permettre d'évaluer correctement les impacts d'un OGM, que ce soit au niveau de sa toxicité ou de son impact sur l'environnement. Leurs craintes ont été relayées par la suite par de nombreux chercheurs, mais elles n'ont jamais été sérieusement étudiées. L'équivalence en substance, peu à peu, s'est imposée comme un dogme religieux. Les détracteurs du concept de l'équivalence en substance se sont vus accusés d'être de pseudo-scientifiques, leurs arguments rabaissés à de la junk science, opposés à la sound science, issue des entreprises ou des universités dont elles financent les programmes de recherche. Le débat scientifique n'a jamais pu avoir lieu, et malgré ces protestations, les entreprises des biotechnologies réussirent à imposer rapidement aux États-Unis leurs semences résistantes aux insecticides et tolérantes aux herbicides systémiques. En Europe, face à la résistance des consommateurs à la création d'un nombre important d'associations qui s'opposent aux cultures de plantes transgéniques et à l'introduction d'OGM dans l'alimentation, les entreprises se sont organisées. L'IFSA obtient, sur un budget global de 12 millions d'euros, un financement de 8 millions d'euros de la Commission européenne pour un programme de recherche Entrance Food. Ce projet avait pour ambition, ouvrez les guillemets, de faciliter la mise sur le marché d'OGM en Europe et donc de placer l'industrie européenne dans une position compétitive. La personne chargée de coordonner cette offensive pour le compte de l'industrie s'appelle M. Harry Kuiper. Aujourd'hui, M. Kuiper est président du panel OGM de l'EFSA et je le salue. Il est chargé de superviser les opinions scientifiques sur lesquelles s'appuient les gouvernements et la Commission européenne pour prendre une décision. Le principe d'équivalence en substance, qui sert de base d'évaluation depuis cette époque, consiste à comparer les composants d'une plante transgénique, protéines, nutriments, antinutriments, etc., avec des plantes conventionnelles. Si les écarts enregistrés correspondent à des écarts connus entre les variétés de la même espèce, la plante transgénique est considérée comme étant équivalente en substance. Un exemple permet de comprendre ce principe est mis en œuvre actuellement par l'EFSA. En mai 2010, l'EFSA a publié une opinion scientifique portant sur une variété de maïs, MON 89034 par 1507 par NK 603. développés par Monsanto. L'agence conclut dans ses documents que les différences observées entre le maïs Mont 89034 et les variétés conventionnelles sont faibles et ne dépassent pas les variations naturelles relevées dans la littérature, ni celles notifiées par la base des données crop composition, mise en place au cours des années par l'EFSA. L'EFSA considère que la comparaison des études fournies par Monsanto avec les données produites par l'EFSA est suffisante pour conclure que la variété transgénique est comparable aux variétés traditionnelles. Les conclusions pour le 1507 et le MK603 sont similaires. Sur son site, Crop Composition de l'EFSA indique à ses utilisateurs qu'ils doivent utiliser, entre guillemets, Ridley and Hall 2004 pour faire référence aux données qu'ils ont utilisées. Monsieur Willy Ridley travaille en fait pour Monsanto. Et note de page, l'EFSA prend soin de signaler qu'il ne saurait en aucun cas être tenu pour responsable des travaux et des résultats obtenus grâce à l'utilisation des données fournies à titre gracieux. En d'autres termes, le panel OGM de l'EFSA se contente in fine de comparer des données fournies par Monsanto à d'autres données fournies également par Monsanto. C'est un cercle vicieux. Je considère qu'en agissant ainsi, l'agence européenne ne remplit pas la mission qui lui a été confiée par ses créateurs en 2002, à savoir garantir l'indépendance, la valeur scientifique et la transparence de ses opinions. Dans la saisine de l'EFSA par la DG Sanco sur les nouvelles techniques de biotechnologie dans les plantes datées du 24 février 2011, il est souligné que, ouvrez les guillemets, le seul objectif d'utiliser les biotechnologies est d'accélérer le processus d'amélioration variétale des plantes de 15 ans à 5 ans, ce qui permet de réduire le coût du développement des nouvelles variétés. Cette déclaration montre qu'au-delà de tous les discours agronomiques ou humanistes, pour les entreprises, l'intérêt final des biotechnologies est de réduire leurs coûts et de maximiser leurs bénéfices. Le 4 décembre 2008, le Conseil des ministres de l'Environnement a demandé à la Commission de lancer une réforme des procédures d'évaluation. Le concept d'équivalence en substance ne permet pas d'effectuer une analyse des risques liés à l'utilisation des OGM dans l'agriculture et l'alimentation. Pour renforcer la sécurité du public et de l'environnement, les gestionnaires du risque doivent impérativement revoir de fond en comble les procédures appliquées par l'EFSA en ce domaine. Et les plantes résistantes aux insectes ou tolérantes aux herbicides doivent subir les mêmes tests que les pesticides produits par l'industrie phytosanitaire avant d'obtenir une autorisation de mise sur le marché. L'EFSA ne peut pas se permettre de réformer un minima ou d'un rafistolage rapide. La société civile et les citoyens qui refusent majoritairement les OGM ne permettront pas qu'une nouvelle fois l'Europe privilégie les intérêts de l'industrie aux dépens de ceux des paysans et des consommateurs. J'ai fait cette intervention qui peut peut-être choquer certains et je m'en excuse, ce n'est pas l'objet. Nous avons convenu avec les responsables de l'EFSA de lancer le débat. Je pensais qu'il fallait le faire de manière claire, de dire clairement quelles étaient mes positions, mon point de vue, qui est celui qui est rejoint par énormément de mouvements aujourd'hui. Et j'aimerais que cette réunion ne soit pas une réunion qui s'arrête simplement dans quelques heures, mais que ce soit le début véritable d'un nouveau débat profond sur l'ensemble des biotechnologies. Aujourd'hui, nous parlons des OGM, mais un autre débat va s'ouvrir dans les semaines et dans les mois à venir sur les nouvelles biotechnologies en agriculture et dans l'alimentation. Il ne faudrait pas qu'une fois de plus, l'Europe soit à la traîne de ses citoyens et de ses agriculteurs et que nous ayons enfin un débat sans tabou où l'ensemble des points de vue peuvent être discutés Et en tout cas, je remercie l'EFSA pour avoir permis ce débat et pour m'avoir proposé de prendre la parole. Je sais que ce n'était pas forcément facile et c'est pour ça que je vous en remercie d'autant plus et j'espère que les personnes citées ne m'en voudront pas. Ce n'était pas évidemment à titre personnel que je les ai cités, mais par rapport à leur fonction et donc ils ne doivent pas se sentir attaqués dans leur personne par mes propos. Je vous remercie.
Dear members of the public, thank you very much for your opening speech. It was very interesting for all of us, I'm sure. Now I'm going to give the floor to our moderator, Helmut. The floor is yours, please.
Thank you very much, Rita. I would also like to thank Monsieur José Bové for his opening and introduction to this workshop. I'm very pleased to be here, and I would also like to welcome you on my behalf to this EFSA consultative workshop on the draft guidance on selection of comparators for the risk assessment of GM plants. My name is Helmut Gaugic. As Rita mentioned, I'm working in the Environment Agency in Austria, and I have been involved since many years in the topic of GMO risk assessment, risk management. So I feel it is a privilege to be here with you today and to have the task of moderating this very important, this very interesting workshop. As it has been said by Rita this morning, I would also like to state from my side that I think this workshop has two main goals. We have a first part, which I think is more or less setting the scene by placing the issue of comparators into the context of the risk assessment framework. We will have introductory presentations and open discussion, among other things, on this risk assessment framework in broader terms and on the concept of substantial equivalence in particular. There is a second part of this workshop, starting before the lunch break, which will focus on scientific issues, and we will have a scientific debate on the contents of the EFSA draft guidance on the selection of comparators for the risk assessment of GM plants. So we have two major goals here, two main purposes for this workshop. The two issues are interlinked, but can also be discussed in a stepwise manner. To some extent, in a differentiated manner, in a stepwise manner, but mindful of the fact that the issues are interlinked. And I think this program gives a very good opportunity to do that. In my point of view, and as a moderator, I would like to share with you my expectations also for this day, and that is, I think at the end of the day, we probably should have a common understanding among our whole group, among these workshop participants, on converging and dissenting views on the topics of our discussion. And when I say on the topics of our discussion, I mean these two major subjects of our workshop. What is important is that your input will be taken into account by EFSA and the GMO panel in the revision of the draft guidance on the selection of comparators, as it has been said by Rita this morning. In principle, my task should be quite straightforward, and as an optimist, I would say it probably is a simple task. I would see my role mainly as being mindful of the timing. I think we have a very interesting, very important program for today, but it is also very dense. So I would like to be mindful of the timing on the one hand, and I also see my role as facilitating the discussion by, whenever necessary, ensuring that presentations and discussion items are to the point of this workshop, to the topics of our discussion, and therefore run smoothly throughout the day. We have a good mix of presentations and discussions. I would like to stress that I would kindly ask all the speakers to stick to the time slots which they have been given. If you do that, which I really kindly ask you to do so, and I will look at that very carefully, then we make sure that we have the ample time of discussion which I think is needed for the topics of today. And my hope and my expectation is that we will reach this good mix and have a successful outcome at the end of the day. So with this brief introduction, I would not like to use more of your time, and I would kindly invite you now to turn to the first part of our workshop. As I said, setting the scene, the concept of substantial equivalence. We have 3 15-minute presentation here as a starting point, and then good time for an open discussion on that matter before we break for coffee. And our first speaker of this morning will be Dr. Sebastian Wu. He's a policy officer at DG Sanco of the European Commission, and his presentation will be about principles of risk assessment in the European Union legal framework. So, Sebastian, you have the floor, please.
Thank you, Mr. Beauvais, dear colleagues from EFSA, dear colleagues from the member states, dear stakeholders. Good morning. I would like first to thank EFSA for inviting me here and to give me the opportunity to contribute to this workshop. Public consultations, stakeholder consultation, scientific consultation is an integral part of the process to develop guidance by EFSA, and we really believe it's a key issue, so we are also very happy to contribute to this process today and to present you the principles of risk assessment as they are laid down in the EU legislative framework. In the EU legislative framework, there are 2 basic legislation. First is the Directive 2018 on the deliberate release of GMOs. This directive is covering 2 types of activities, basically. Cultivation and also what is referred to in the jargon as important processing, which means in practice placing on the market of GMOs all activities related to GMOs besides cultivation. The legislation is clear. Prior authorization of GMOs, you need a thorough environmental risk assessment of these GMOs before considering the authorization. The second piece of legislation, basic piece of legislation, is the regulation on GM food and feed, which provides for an integrated approach for the whole food and feed chain. So when it's a product that is intended to go in the food and feed chain, in this regulation, you cover both the cultivation activities, but also the placing on the market on food and feed, as well as other uses, which are referred to as important processing for other uses. Under this regulation, 2 times of safety assessment has to be carried out. The first one is environmental risk assessment for all products who contain GMOs, and this environmental risk assessment has to be carried out in accordance with the principles set out in the Directive 2018. And the food and feed risk assessment has to be carried out for not only products containing or consisting of GMOs, but also of food and feed that are derived or produced from these GMOs. Now moving to the principles of risk assessment. So first, in the Directive 2018, there are quite detailed requirements already that are existing in the directive, both in one annex, which is Annex 2 of this directive, and in a decision which was adopted in 2002, which is complementing this Annex 2. The annex is first describing the objective of the environmental risk assessment, which is to identify and evaluate potential adverse effects, either direct, indirect, immediate or delayed, with a view to identify which are the needs for risk management measures, that are needed. There are 4 general principles that are set out in this annex for the environmental risk assessment. The first one is that the new characteristics of the GMOs have to be identified and assessed, and to compare these new characteristics with non-GM counterparts. The different use of the GM modes that would be induced by these new characteristics needs also to be assessed. And it is underlined in this, on this topic that it is key to define a baseline, the reference where one has to, on which we have to base this assessment. Second principle is that the assessment has to be scientifically sound. It has to be based on common methodology for the assessment, and one has to characterize the uncertainty which is associated to each methodology that is used. It has to be case by case, which means that it has to be tailored to the GMOs, the GM, the recipient organism, the trait, but also to the receiving environment. And last element, it has to take into account new information when it becomes available. The methodology for the risk assessment is also laid out, and you have this, this is taken out of the decision of 2002, with methodology in 6 steps, which are also detailed further in this decision, which is and that are here. First, hazard identification, hazard characterization, risk assessment. And when these 3-4 steps are carried out, still 2 additional steps, which are to consider what are the additional risk management measures and strategies that can be developed prior to finalize the overall risk assessment. Final aspects of these requirements, it also sets out the conclusions and the points on which the risk assessment has to conclude. 9 points are detailed in these requirements, which are just summarized here. Points are related first to the behavior of the crop of the GM as such, so aspects related to persistence, invasiveness to the environment, the risks of gene transfer, the impact on target and non-target organisms, as well as possible impacts on the biogeochemical processes, and then human and animal health. Moving to the food and feed safety assessment. So this is handled in the regulation on GM food and feed. Under this regulation, the basic requirements are much less detailed than in the directive. So what is laid down in the regulation is that food and feed must not have adverse effects on human health or animal health and the environment, not mislead the consumer, and have differences that would be nutritionally disadvantageous to the consumer. The regulation also provides for the definition of the conventional counterpart, which is a similar food and feed produced without the help of genetic modification, and for which there is a well-established history of safe use. Now, moving to the safety assessment as such, the next 2 slides are not set out in the regulation, in the legislative framework, but are set out in the guidance of EFSA and in the draft regulation of the Commission that is in preparation, that will be adopted in comitology. Here the strategy is that one compares the non-GM with the GM, but first the first thing in the safety assessment is to focus on the modifications and to carry out an assessment of these modifications, either the intended modification as such, so the safety of a new protein that is introduced, but also the non-intended modification that would be resulting from this modification. The second aspect is to assess the safety and to compare the non-GM with the GM to assess that there are no other modifications that have been brought in the GM plant, so as to identify possible additional modifications and to submit them as well to a thorough safety assessment. If we look at this in more detail, so we have first the assessment of the intended modifications, where we look at both the toxicological, the nutritional, and the allergenicity aspects. This is made mainly based on the molecular characterization and the comparative analysis. And then in the second stage, you got the assessment of the whole crop to either confirm the absence of modification or identify other modifications that are then submitted also to a thorough safety assessment. In accordance with the guidance of EFSA, there could also be need for tests with whole food feed. This has to be decided on a case-by-case on the basis of the outcome of the preliminary of the previous steps of the safety assessment. This is certainly an issue that is still under discussion, at least in the process of the regulation that the Commission is now preparing. The last steps of these safety assessments are then the exposure assessment and the risk characterization. What is set out in the regulation, and which is important for the safety assessment as well, are the aspects related to labeling. And there is a requirement, as you know, to always label products containing GM or produced from GMOs as genetically modified. But there are also additional rules that are foreseen in specific cases, and it is where the GM food or feed is different from its conventional counterpart with respect to its composition, nutritional value, intended use of the food, or implications for the health of certain sections of the population. As I already referred before in my talk, the Commission is now developing more detailed guidance through comitology. So for food and feed safety assessment, our objective is to finalize the process in 2011. And for the environmental risk assessment in 2012. The objectives are basically the same for these two processes, although as I outlined, the general framework is a bit different. And it is to provide more detailed requirements, for example, by including protocols that would be requested for certain studies. Through comitology, to the comitology process, increase the sense of ownership of member states, since they will, and we are discussing with them these aspects before we submitting to the vote in the regulatory procedure. And by doing so, also improve the scientific and legal certainty for, I would say, EFSA, but also the applicants, so that they are clear on which are the elements that have to be provided in the applications. So just to conclude, it shows that we have a comprehensive risk assessment framework in the EU with different layers and different actors involved in these different aspects. We have first requirements that are set out in the basic legislation, which is adopted by codecision, and the Council and the Parliament are involved. Then we've got a second set of requirements that may be adopted by comitology, and there it is the Commission in collaboration with member states which are involved in the formal process. And thirdly, and the more detailed requirements are set out by EFSA in the guidance. These guidance are developed through public consultations, so I believe it's closing the loop because it's why we are here today. And with this, I thank you all for your attention and to this thing.
Yeah, thank you very much, Sebastian, for your very clear presentation and for sticking exactly to the timing. That's very helpful. My suggestion is that we hear all three presentations of this morning's session and then enter into a discussion, unless there is one urgent question of understanding. I would allow that, but please, discussion. We enter into the discussion once we have heard all the three presentations. Is there an urgent question of clarification, understanding? That does not seem to be the case. So once again, thank you very much, Sebastian. We move on, and I would kindly invite our next speaker. This is Dr. Hartmut Meyer, who is the scientific coordinator of the European Network of Scientists for Social and Environmental Responsibility. The title of his presentation will be GM Plant Environmental Risk Assessment, Pros and Cons of Different Approaches. So Hartmut, you have the floor.
Yes, thank you very much, Helmut. I also would like to thank EFSA for the invitation and José Bové for also taking the initiative to launch this really important discussion. And actually, it also came a bit to a surprise that after such a long time, we again have to talk about the substantial equivalence and familiarity principles. We're happy to do this. And I would like to concentrate on the environmental risk assessment because as the legal situation is constructed with this one door, one key approach, many scientists have a slight fear that the concepts and principles of environmental risk assessment are not exactly reflected in the approach of the food safety assessment. Actually, it's just starting with the wording. It is a safety assessment on the one side and a risk assessment on the other side. And that is not just different words, but could also be different principles. Just to go very briefly into the history, and I don't want to take away your points, I just have to quote a bit of the OECD work. The concept of substantial equivalence has a long history, and it has been debated controversially, as we have heard in the first opening statement of the director of EFSA. What it actually means, and we have this 1993 publication, is that there are, in general, three principles. The first one here is if a food is declared to be substantial equivalent, and there are no concerns are then expected in the following, that essentially means no risk assessment is actually needed for such food. The second case is when a product is determined not to be substantial equivalent, then there is a need for further evaluation. And the third case is, and this is also a matter of our discussion in the afternoon, when there is no competitor available for whatever reasons, then the old paper of the OECD suggests the food should be evaluated on the basis of its own composition and properties. These are the three principles that apply. The second concept, which always pops up in this discussion, is the concept of familiarity. So it is not the same, but at least in my understanding, it has been developed to exactly deal with the case number 2. So what happens if there are differences between a GMO and its conventional counterpart? Then the concept of familiarity mostly comes in and tries to evaluate if these are actually unexpected and very new differences, or if we are familiar with those differences. What does this mean?
if we are familiar. I think, and here is also the link of these concepts to the topic of our workshop today, to the second part as well, which is the topic of competitors. To what kind of organisms or what kind of food are we going to compare the GM food? A paper of the FAO and WHO from '96 elaborates a bit on that and comes with the conclusion that we have to look at the natural range of variation in competitors. There was a long discussion about which competitors are the appropriate ones, and again also a OECD paper from '93 gives a bit of guidance. What does this mean in the context of environmental risk assessment? What are the competitors? And at that time, it was concluded, amongst others, that it could be lines of the crop plant developed with more traditional techniques or other plant lines developed by the same technique. Essentially, that means if we have a GM maize plant under assessment, we could compare it with any other GM maize plant to see whether the differences caused by genetic engineering are decided to be new and subject to risk assessment, or if we can claim that we are all used to this difference, or nature is used to deal with these differences. There has been a lot of criticism on this concept, and I just want to quote one of the quite well-known papers from '99 from Milstone et al. And I think this is valid until today, and in the EFSA guidance and documents we find some attempts to overcome this criticism in a way. The concept of substantial equivalence has never been properly defined. Actually, and I think this is also what is a reason of all the trouble and all the concerns, the concept of substantial equivalence has been developed in a very different legal environment than the EU. It has been developed in the concept of the US approach, that there is no specific regulation necessary, and essentially together with the concept of familiarity, it has been principally developed to not conduct risk assessment, but to declare the effects as safe, which are caused by the application of the technology. This approach is, in a way, opposed to the European legal system, and in my point of view, this is one of the major cause of all the problems we face with this adoption of that concept. Also, in the field of environmental risk assessment, there have been a lot of criticism. I quote a paper of Perry et al., and before I knew that the author was here, so forgive me. And it also says that the concept of a history of safe use, as it comes from food safety, relates less easily to the environmental risk assessment, and that the broad use of whatever competitor is there, the extraneous varieties, as it is noted in this article, doesn't make practical sense for environmental risk assessment. And I think we need to take these criticisms into account when we are going to develop future guidance, especially if these guidance are then turned into a legally binding document. And I just want to remind you that the concept of familiarity has never been recognized in the legal concept for GMO environmental risk assessment. It has been brought into the discussion of the Cartagena Protocol in '96. It was discussed in the working groups 2 and 3, and it was rejected in the working group number 4. You won't find a reference to the concept of familiarity also in the Directive 2001, which is the legal framework for environmental risk assessment in the EU. Those environmental legislations are quite explicit on the choice of comparators. Both say that the non-modified recipient or parental organism in the likely potential receiving environment should be served as the comparator in the first place. That is the language of the Cartagena Protocol. The 2001-18 has a comparable language. So this also says that the broad concept of familiarity is not acknowledged in these legally binding documents. What was the outcome of all this international and European discussion? The outcome is that the Codex Alimentarius, which is the first code, and also the European Commission and its food and feed legislation says that substantial equivalence is the start of everything, but it's not a safety assessment in itself. That leaves the question, what is the safety assessment then? What do we do? And here I think the guidance, also together with the other documents which are available from EFSA, still leave a certain ambiguity. We can discuss if this is just language, if this is intent, or if this is a misunderstanding, but we have to go through this, especially when it comes to a legal binding text. It says, for example, that these statistically significant differences between the GM plant and the competitors have to be assessed if they are biologically relevant. And only when they are judged to be biologically relevant, they go into the risk assessment. This, of course, to a certain extent makes sense. It might be debatable if we really should assess all differences which are there. So a decision on relevance might be useful. But of course, we need to know how this decision will be taken. What are actually the limits of concern? What are the parameters on which EFSA in the end is going to measure relevance? And what at least I found a bit disturbing in this context is then in the second part, in this context, EFSA in the guidance documents, and that is also quoted in the comparator documents, again quotes the concept of familiarity. I think this may cause some concern and troubles, and we have to really discuss about this crucial issue in the whole concept. There have been a lot of questions reading the text. If EFSA, and this is also, going to mark it now, if EFSA is just simply renaming something which has been, at least for environmental risk assessment, rejected quite a long time ago. The comparative safety assessment will make crucial decisions for environmental risk assessment. And at least from reading the text, the people who have written the comments for the ENSA network were not really clear and were not convinced that that was the right approach. The recommendations which I would like to put on the table is that the concept of comparative safety assessment and of familiarity as applied prior to the environmental risk assessment should be abandoned. We find that in the 2018, of course, it contains a principle that says you must compare the GMO with the parental organism, but this has to be done in the environmental risk assessment. I think this we should stick to. And also, we should have a good discussion on what is going to happen when there is no appropriate parental organism. The EFSA document suggests there could be a risk assessment per se that would also be in line with the old OECD document. And it is really a debate what we do with these kind of organisms where we do not have an appropriate comparator. It will be very important for our future discussion and also very important also for the acceptability of future decisions that there must be an establishment of the scientific criteria for judging these significant differences between the GMO and the biological comparator. Because also in environment, you are not only to talk about the relevance for food safety, you need to talk about the relevance for environmental risks which I guess is a much broader field than just comparing the constituency of an organism. You need to compare the behavior. And something which, from an environmental perspective, is very important is that there is a second set of comparators. It is exactly the environment in which you are going to compare your GMO and its competitor. Because to establish if something might cause harm or not, you need to have a sound decision-making process on which system you take as a competitive system. Are you going to compare the BT maize against an agriculture environment with a high use of insecticides, which is sort of the high end of industrial agriculture, or are you going to use an environment in agriculture which makes use of much less or no insecticides? These are very crucial questions on the side of the environmental competitor, which also has to be clarified in future. Just let me conclude with a short quotation from Commissioner Dalli. He made in March 17 on the last meeting of this kind. And actually, he spoke about the confusion with the term substantial equivalence. He brought back the language of the comparative approach, which, at least when we follow all the documents, was the language of the previous EFSA guidances. So the competitive safety assessment as such as a new principle actually is something which he at least did not really mention in this speech and this part. And Commissioner Dalli also suggested that all differences which can be detected need to be assessed in further risk assessment. That, at least in my interpretation, would end the debate on the concept of substantial equivalence and the comparison. So I think we have to see what actually is meant by these statements. And ANSA is determined to engage critically and constructively in the further debate, as we have done so in the past. And I hope that we get a lot of input and suggestions during this meeting. Thank you very much. Thank you very much, Hartmut, for a very clear presentation. And again, thank you very much for sticking quite well to the timing. This is very helpful. Again, I would like to suggest that we hear the three presentations of this morning first before we enter into the discussion, unless there is an urgent question of clarification or understanding, which does not seem to be the case. So thank you once again, Hartmut. I would now like to invite our next speaker, Dr. Peter Kearns. He is Principal Administrator at the Organization for Economic Cooperation and Development, the OECD, in the Environmental Health and Safety Division, and among other things, dealing with biotechnology and biosafety there. So, Peter, you have the floor, please.
Thanks very much, Helmut. Good morning, ladies and gentlemen. It's nice to see you all. Many old friends, but some new friends also. Thanks to EFSA for inviting me here. I'm not going to try and do anything too extravagant this morning. I just wanted to explain a little bit about this famous concept of substantial equivalence from a historical perspective. Very much of this will be my personal ideas. How does this work? Yes. Okay. This is a complicated slide. Many of you will know all the details here, but all I want to show by this slide is that 1993 was when OECD published its Green Book on the concept of, including the concept of substantial equivalence, which we've already heard a little bit about. And 1993 was quite a bit before we had actually any commercial uses, particularly from the perspective of food and feeds in terms of transgenic plants or transgenic foods. What the people who worked on this were trying to do in 1993 was to figure out how they were going to deal with these regulatory people who have the responsibility for food and feed safety. And they were trying to think at that time, how do we manage this issue? How are we going to manage this issue? We know that we're going to have these dossiers coming on our desks very shortly. What are we going to do? How are we going to do a credible safety assessment? But this was very proactive. Nobody had actually looked at a dossier before, while they were trying to develop the ideas of what they were going to do when they actually had to see one. Here in 1994 was one of the first things. It was the famous Flavor Saver tomato. And that was done rather differently. That was in the US, and it was, if I remember correctly, a food additive petition. But that came after the concept was developed of how people, regulatory people thinking together, how are we going to deal with these things. 91, I'm just mentioning this for historical reasons. In 91, there was work by FAO and WHO, which began to look at strategies for safety assessment. This was followed in 93 by the OECD book. We've already had a slide about what the concept of substantial equivalence is. I've got three slides here. And one of the issues to do with language from the Green Book is, if you read this, I leave you to take home my slides and read this, or you can read the whole book. It's very turgid language when you read it in retrospect. It's a little bit difficult to get your head around all these convoluted words. But one of the things it does sound like, if a new or modified food component is determined to be substantially equivalent to an existing food, then further safety or nutritional concerns are expected to be insignificant. It sounds like an endpoint. It sounds like a decision to be taken. And what happened subsequently, after this turgid language was developed, is people figured out that it wasn't really an endpoint they were talking about. It was more a kind of guiding principle about how you do a safety assessment. More turgid language. Take it home and read it. To be fair, when we worked on this book in 1993, I'd said there was no commercial applications, and there hadn't been commercial applications, but you will find examples in the book where people go through case studies and think, based on certain case studies that we know about, it mentions the potato there. People already had viral resistant strains of potato, for example, some of these other things, but none of this has been commercialized. You have here also the mica protein. That was something that was quite different, but an example of different examples of how you might apply this concept in practice. FAO built on the work that we did in 93, and here you begin to see language which is talking about a dynamic analytical exercise in the assessment of the safety of a food relative to an existing food. This is Harry's table from an article Harry wrote in 2001. The concept of substantial equivalence is a starting point for a safety assessment. It involves a comparison between the GM organism and its closest traditional counterpart. It allows you to identify intended and unintended differences on which further safety assessment should be focused. It's not surprising that you see this concept evolving as people are actually having to do risk assessments, safety assessments in practice. This is no longer a theoretical exercise of how we're going to work together. It's a concept that by this time is being put into practice. And this is the emergence of the comparative approach and how the concept of substantial equivalence is implemented in practice. I just put this up here just to show the following. Harry's paper in 2001. I was also involved in a similar exercise in 2002. This is just me showing off a little bit. This is the Codex guideline, which comes in 2003. By this time, we're not seeing so much substantial equivalence cropping up in these types of documents, but we see here again this comparative approach should be compared with an equivalent analysis of a conventional counterpart grown and harvested under the same conditions. Why did I put that? Okay. This is a little bit about the work that we do at the OECD today. And this is a little bit about how we implement the concept, the comparative approach today. What we tend to work on is crops, foods derived from crops, which are transgenic. And what we do in these documents is we have, this one deals with cassava, which is an important crop in Africa and other developing countries, very prone to viral infections and viral attack, and people want to make transgenic varieties of this thing. So what we do in documents like this, for example, with cassava, is we put together information. By we, I mean OECD safety, people involved in safety assessment, and we compile information. We've done this on many of the commodity crops, soybean, maize, and so on. Here we put together those important components on a crop-by-crop basis, and we look at what the composition is for nutrients, anti-nutrients, toxicants, and allergens. So that in one compilation, you have what, in the conventional counterpart, if you like, are the major components and at what levels they are present in the particular crop. And then you have information on which you can compare with a new variety. So basically what I've told you here is, or tried to explain, is that this is a dynamic concept that has evolved over time. It means a very different thing today from what it did, I think, because of practice, because people have had to do safety assessments over the years, and so the application of this concept has evolved with time. And it's about the comparative approach.
Thank you very much, Peter, for your very clear presentation. Excellent timing. So this gives us now the possibility to enter into a discussion among ourselves. We have had 3 very clear presentations, setting the scene, outlining broader concepts into which risk assessment practice is enshrined, and now is the possibility up to the coffee break to discuss. What I would like to do is to move up there, so in order to be in a better position to facilitate this discussion, this is upon the prerequisite that I may borrow every now and then Sebastian's headphones. I like the French language very much. I speak and understand it a little, but probably not good enough to follow a complex discussion. So, dear colleagues, about half an hour time now to discuss. I'm asking who would like to take the floor. Yes. Joe Perry, please.
Thanks. I've got some clarifications that I think would be useful regarding Dr. Mayer's talk. First of all, I'm very pleased to clarify that there is no decision-making prior to an environmental risk assessment. If you read the guidance document, the point we're trying to make there is that there is some data which comes from molecular characterization or from compositional trials. In other words, it comes from issues concerned with the molecular characterization and food feed guidance document. So there is some data which comes to us which is relevant to the ERA and which must be considered before we even start. And thereafter, we go through steps of problem formulation. Those steps will indicate the need for further data, which is then gathered before the overall environmental risk assessment. So there's no, I'd like to reassure you that there's no question of EFSA suggesting any new comparative approach or that the comparative approach should terminate prior to the environmental risk assessment. Indeed, the comparative approach is central to the ERA, and I think the guidance document is clear on this. If you still feel that there is scope for misunderstandings, if you would like to set out exactly what those are in a letter, then we'll certainly be very interested to have a look at them. My second of 3 points relates to your recommendation that scientific criteria should be placed for statistical significance. In fact, this is now done both in the food feed guidance and in the updated ERA guidance through the equivalence test. Particularly for, you mentioned ERA, here the equivalence limits relate to actual environmental harm and they are derived from protection goals. And this is really a way in which we are quantifying and formalising this concept of substantial equivalence. And it's done both for food feed, there's an equivalence test for food feed, and for ERA. So in both cases, we are attempting to formalise and scientifically quantify this concept of substantial equivalence. With regard to your third point about the comparison, the 2001-18 principle that GMO should be compared with parental organisms, you will see in the Comparators Working Group draft document that this is the case for singles, that we recommend the use of conventional comparators for singles, and indeed when we evaluate stacks, that data on the singles is available to us because for stacks each of the singles is evaluated before the stack. So the parental data is there, and that's the case whether or not a conventional counterpart is available for the stacks or not. In some cases for ERA, it would not be sensible to stick slavishly to the principle of only using parental comparators. Let me give you an example. Drought-tolerant crop. If you had a drought-tolerant crop and you tested it in conditions of extreme dryness, your conventional counterpart may well die. This would not help the assessment. Therefore, in ERA, we allow ourselves the ability to look at additional comparators where this is sensible to answer specific questions, and I'll develop that more in my talk. So there is no problem for the ERA guidance document to develop a risk assessment per se.
Okay, thank you very much, Joe, for your contribution, intervention. I kindly ask those who would like to intervene to give me a clear signal by hand. And once you take the floor, please just use the microphones which you have in front of you and mention your name and affiliation so that we all know who you are and where you are from. And then in your intervention, please be brief and concise so that all can benefit from the discussion. My suggestion is that I collect a few comments and interventions just like now, and then give the possibility for our colleagues on the podium to respond, and we see how it goes, and then may enter into a more multilateral discussion. So the next to ask for the floor is our colleague here at the back.
Yeah, thank you. My name is Stefan Rauschen. I'm a researcher and lecturer at RWTH Aachen University in Germany, and I'm also the Executive Secretary of the Public Research and Regulations Initiative, PRRI. I've been working for more than the past 5 years in environmental risk assessment research in Germany, funded by the German Federal Ministry of Education and Research. And so I would like, from that perspective, comment on a few things that Hartmut mentioned in his talk, which basically go in the same direction as Joe Perry just mentioned. What we do in our field release experiments, and we performed 2 of those over the last 6 years in 2 different receiving environments, is that we compare the genetically modified variety that we are looking at with its near isogenic parental line and also conventional varieties. And what we found is that conventional varieties may exhibit not only statistically significant differences in the occurrence of non-target organisms or the impact on microorganisms in the soil and similar things, but these differences are actually tremendously large and much bigger than those that we have observed between the genetically modified variety and the near isogenic parental line. And so the question arises, actually, if conventional varieties can be regarded as being substantially equivalent in their interaction with receiving environments. And if we, in fact, say that conventionally bred varieties are safe and are acceptable in the way how they interact with the environment, then the question is, how do we range in, how do we use and assess those variations and make them usable for the environmental risk assessment of genetically modified plants? And as the scientific results are at the moment, all the genetically modified varieties that we currently have on the market and that have been assessed very thoroughly also by EFSA have no impact or no effect on the receiving environments that go beyond the effects that we see with conventional varieties. And this means basically that we cannot make an environmental risk assessment or any kind of risk assessment without appropriate comparators. And then it very much depends on the questions that you are looking at, and that basically determines and drives the choice of the comparator that you need to use.
Okay, thank you very much. Beatrix.
Thank you very much. My name is Beatrix Kapados. I'm the Federal Agency for Nature Conservation in Germany. I would like to focus on 3 points. The first is that with a comparative approach with a concept of substantial equivalence, which should be a starting point of assessment, we are mainly focusing on quantitative aspects or not on qualitative aspects. So I like to exemplify that, and that may be on the science side, that may be a shortcut, especially when it comes to environmental aspects, but not only in the context of environmental aspects. And as an example, I like to quote or focus on prions, which cause bovine BSE and Jakob-Creutzfeldt disease in humans. Looking into the amino acid components of these proteins, they may be no difference, but because of the different configuration, there is a big qualitative difference. So what I like to focus on that with that point is that we somehow, because we are interacting with complex environments, that we need really some form of intelligent bioassays to have, in addition to the quantitative assessment, some form of qualitative information if what is in the quantity is also in the quality. So that is the first point. And in that sense, the OECD consensus document and the food and feed guidance is And in the food and feed guidance, there are some basic aspects which are also relevant for the environmental assessment, and that is the molecular characterization and the compositional analysis. That is done also for the environmental risk assessment according to the food and feed safety assessment. But that may, for the environmental risk assessment, there is an interface for sure, but that may be not enough. For example, if you have a potato, the potato as such will be evaluated because that is our food, our feed component. But we do not have this form of information on the whole plant. And the plant, the leaves or whatsoever is what is interacting with the environment. So there may be the need for having a different additional information in the context of the environmental risk assessment. And my impression is that that has been not really put into account, at least enough, in these both documents. So that is the first point. I try to yes. A second is in the context of the comparison. And I would like to emphasize that really once again, because I think it is a very important point. What is our comparison? Which agriculture is our comparison? We had quite a lot of exchanges about that, but I like to underline that once again, that it is very different in different countries, and especially under the biodiversity aspect. And we do know that we have biodiversity losses. We do know that we have political goals on stop the loss and things like that, and we have to take that into account when we define what is our agricultural comparison in this area, and I think there has not been enough discussions about that. Thank you.
Okay, thank you, Beatrix. I take one more intervention before I then give the possibility to our colleagues on the podium to respond if they would like to do so. So, Susanna, please.
Thank you. Zsuzsanna Bardos, Ministry of Rural Development in Hungary. I would like to make 2 or 3 comments. One, I can't see the examination of the delayed effects, neither in the environmental nor the food and feed risk assessment. With proper risk assessment, if you have got a proper experiment, only one thing should be changed. Therefore, the Codex Alimentarius way of expressing things, that the control should be grown and harvested under the same conditions, would be essential. And this is not in the ERA guidelines. For the comparators to use any commercial line, it occurs to plant biologists or people working with plants, it appears. To an animal experiment, a person would never occur to compare, let's say, a hooded lister rat with a Wister or a Norwegian brown, or a Highland cattle with an Aberdeen Angus or a Holstein or anything. So I think that the only proper comparator is the parent line, and I can't imagine a situation when there is no proper parental control. Thank you.
Thank you very much. Now we have heard very clear and interesting contributions from the audience. Is there anybody on the podium who would like to react to some of the issues raised? Because if that is not the case, I would like to continue to give our colleagues the possibility to raise comments, and please give me a signal if there is any need from your side to react. You already had the floor. I'm looking if somebody else would like to come in. Inge, please, and then Pete.
Yeah, thank you. Inge Bohr from the University of Rostock in Germany. I just have a question of understanding to Dr. Meyer, because as far as I understood it, he said if there is no comparator there, we need an environmental risk assessment per se. And this is something I absolutely don't understand. So what does it mean? In detail, what should we do then? Adding to the question that I can't understand how there can be no comparator at all.
Okay, this was a very clear question. I don't know if Hartmut, you would like to respond, because then I would like to give you the floor now, and then we move on with Pete on the understanding that you would like to raise a different point. So please, Hartmut.
Yes, thank you. As I said, we should explore this idea. It has been mentioned in the paper of the EFSA as such, what I understand as an idea or concept. It has not been elaborated in detail in that paper. And it comes a bit close to the third principle of the old OECD paper that if there's no comparator, you need to assess the foods as such. I think such an idea would come as an alternative to the suggestion to take a broad range of comparators. And I think we might enter into a discussion how this could be done and if that is really an alternative to other suggestions. So there is no concept behind that. It is more or less an idea.
Okay. Thank you very much. Please.
Thank you, Chairman. My name is Piet van der Meer. In my former life, I've been the competent authority for the Netherlands for GMOs for over 10 years. I have been deeply involved in the OECD guidelines, the EU directives, and the Cartagena Protocol. The last 10 years I have an easier life. I teach about biosafety and I provide assistance to governments and organizations, one of them PRI, with which I'm here today. First of all, when we developed in the very early stages, and I'm talking about 1987, 1988, the basic concepts for biosafety assessment, risk and safety assessment, the concepts of familiarity, comparative, substantial equivalence, those are very sound concepts, and I've not seen any argument why those would not be valid anymore today. The first question that comes up with risk assessment is what do you compare this with? So the comparative risk assessment, however you turn it around, you have to compare it with something. That's the first part. The second part, familiarity, is the simple question, what do we know about a certain impact? Insect resistance, have you any familiarity with this? Are there similar crops available? So we do use this concept of familiarity. Just to correct the point that Hartmut made that this was rejected in the Cartagena Protocol, no it wasn't, it's incorrect. I have shared that part of the negotiations. And familiarity is very strongly embedded in the Annex 3 in the risk assessment. It's not used, the word familiarity, but that as such doesn't mean that the concept is not used. It very clearly says that any identified risks are compared in the context, not even with, but in the context of similar risks with the non-modified comparator. And then a more general point, I think we should be very clear, because if you produce, every farm breeder knows this, if you produce new varieties, you will find after every crossing a lot of changes. And we should be very clear that first of all the changes we see, not every change is significant, and not every significant change is relevant. You see a lot of changes in producing plants, where you use conventional breeding and genetic modification. And we should be very clear, because the basis of the annex in the protocol and the directive says you have to identify changes linked to the genetic modification. And here comes the problem, because very often you see a lot of changes, you have no idea where they come from. And to address this kind of issues, complexities, the concepts of familiarity, substantial equivalence were brought forward. It's not a risk assessment in itself, it is a trigger, it's a tool how to go forward in the risk assessment. But I think we should bear in mind that not every change is significant, and you should see them in the range of what happens in a variety, and that not every significant change is relevant. If you suddenly see that plants are green, well, they are always green. You have to ask yourself, is there any relevance to safety in this context?
Okay, thank you very much, Pete. Any other reactions from the floor? Yes, Stefan.
Thank you. Stefan Eklöf, Swedish Board of Agriculture. I just wanted to comment on an intervention that was made about insufficient information in dossiers for GMO risk assessment. And the opinion was expressed that there was information mentioned missing. And one example was then analysis of leaf tissue, for example, vegetative tissue, in analysis of a potato. However, I would like to point out something that has to be very clear for everyone that listens, and that is, well, I work as a risk assessor, and many of us do, and that we can request just any information. There are no restrictions, and we do. And that has to be known. Thank you.
Thank you very much. Any other requests for the floor? You already had, but okay, please, Joe.
Joe Perry, GMO panel. Just a couple of clarifications. Someone, I think it might have been Dr. Tapeza, mentioned the importance of looking at plants in the environmental risk assessment. I can reassure you that that is fundamental indeed. Reading from section 2.1 of the Comparative Safety Assessment of the ERA guidance document, it states, when looking at GM plant-environment interactions, in-planter data are the fundamental source of information. So we certainly agree with this. Replying to Dr. Bardosh, I've already given an explanation of where a parental line would not. From a scientific point of view, I'm not talking about the legal point of view, I'm talking about science. For good science, it is sometimes not sensible to use a conventional comparator. Another example, if you want another example, would be GM fish. I know we're only discussing plants today, but GM fish which have been bred to be cold tolerant, one would not wish on, not only on scientific grounds, but on health and safety grounds necessarily, to compare them with a conventional fish in an extremely cold environment.
Thank you very much. Please, Stefan. Yeah, thank you for giving me the floor once again. I just wanted to mention two studies from the recent literature which I found quite interesting for the discussion of today. One is there was a study recently published by Anna Koll and some colleagues who looked at the genomics and transcriptomics of Monet 10, the very highly contested BT maize variety, in different genetic backgrounds. And what they found was that indeed the differences between the different genetic backgrounds of the varieties were much more different than differences between the GM variety and the neoisogenic parent. So this again shows, and there have been some other examples for that, that differences between conventionally bred varieties can be statistically significant and can probably also be biologically significant, and we need that kind of assessment to actually assess whether a GM variety is in any way different from the whole spectrum of variation that we find within the varieties that we currently have. The other example was actually more interesting for me as an entomologist, because what colleagues did was they looked at different clones of a naturally occurring grass in Europe. And they took those different clones, took them to the greenhouse, took aphids, put them on these grasses and let them thrive there for some time, and assessed how those aphids performed on the different clones of that naturally occurring grass. And they found differences in the performance of those aphids. And what they then did was to see whether those differences actually manifested and carried over to the next trophic level. So they looked at the green lacewing, which is a predator of aphids, which is highly important as a biological control agent. And they found that those different naturally occurring clones had impacts on the green lacewing. So this again shows that even in nature, without any human intervention, we have large differences, which can also be of ecological relevance for species that we as humans value because they have very important ecological functions. Taking that in mind, having that in mind and taking that into account is crucially important for this whole discussion that we are currently having. And I really urge people to look at those kind of studies that are coming in scores in the published literature over the past years and months, which are of high relevance for that. And I congratulate EFSA on taking those studies into account in their scientific opinions. Thank you.
Okay, thank you very much. We are slowly but surely approaching coffee break, but before we do that, I would like to ask if there is anybody on the podium, any of our colleagues who would like to react to some of the issues raised. If that is not the case, any urgent requests for the floor before we close this part of our meeting? Harry and then Pete.
Thank you, Chair. I was very pleased with the presentation of all the speakers, and in particular Peter Turns, who emphasized the issue of the evolving aspects of the substantial equivalence principle. I think he's right. The way it has been defined years ago in the 70s, 80s, and compare it with the situation now, how it is applied, that makes a lot of difference, a hell of a difference, I would say. Peter mentioned some of these OECD documents, the very first ones on GM crops, didn't even show a compositional analysis. Now, if you go to an EFSA document now and look at the chapter on the compositional analysis, there is no comparison with earlier days. So I think, and that's my question also to Mr. Beauvais, who gave a very interesting presentation and criticizing the principle. It would be good to get some feeling from this audience. Is the comparative approach, is that a workable, good working principle for risk assessors, as EFSA does it? But not only EFSA, also other agencies apply this approach. There is one other aspect, besides the comparative aspect, and that is the fact that we may pretty soon be faced with products for which it's very difficult to find a comparator. That's also in this comparator's opinion. But still, you have to do a risk assessment. And I want to emphasize that in terms of human safety, there are a lot of chemicals to which humans are exposed, which can be highly toxic. And there is no comparator there. But still, we should be able to assess those products as well. So also the area of non-comparators is an important one. So I would say both. It's the comparative approach if you have an appropriate comparator. If you don't have an appropriate comparator, we still should be able to do a proper risk assessment. And I hope that Mr. Beauvais could comment on that we have a sound working principle, yes or no.
Thank you, Harry. Pete?
Thank you, Helmut. Much has already been said by Harry that indeed these concepts are being continuously fine-tuned. I think it will be good to mention in this audience that under the Cartagena Protocol, currently we are discussing this in a group called the Ad Hoc Technical Expert Group on Risk Assessment. Helmut is part of that group and other members in this group. We are currently refining these things. But as Harry said, the basis is a comparison. You have to make a comparison with something. And I believe that the EFSA has done a wonderful job with very robust opinions over the years. I believe they are very well independently verifiable. There's no problem with those. One comment I'd like to make in reaction, one of the interventions was made, we can ask for more information, and we do. There we should be very clear that as government officials should only ask for more information if it's relevant to the risk assessment. And to repeat a golden rule of John Berenger from ACRE many years ago, only ask for more information if you know what you will do with the answer. Because that's what we see happening at the moment as a sort of a safety net. We keep just asking and asking more information. But to be very clear, this is in the interest of risk assessment, and additional information asked should be relevant. And what we do note in the EFSA guidance is going from very robust opinions, very clearly stated, to guidance that seems to be asking more and more and more, even if without a specific risk-related aspect. The idea of if you have something, give the information. And let me just underline is that safety is not helped by being flooded with information. As risk assessors, and I've been done this over 25 years, it is difficult enough to find your way through. And if you get flooded with information that's not entirely relevant, it doesn't help safety. Thank you.
Okay, thank you, Pete. As I said, we are slowly but surely approaching coffee break. Are there any final requests for the floor? Monsieur Beauvais, s'il vous plaît.
Oui, juste pour répondre en quelques mots à M. Cooper qui m'a interpellé au même titre que le panel sur la question de l'évaluation des risques. Je pense qu'effectivement aujourd'hui le débat il se pose de cette manière-là, de manière différente, je suis d'accord, et qu'aujourd'hui je trouve intéressant d'ailleurs que dans le projet de la commission, du commissaire Dalli, sur les nouveaux éléments qui permettraient à un État d'interdire ou non la culture, un certain nombre de points nouveaux sont pris en compte. Et je crois que justement ça fait partie des nouvelles évaluations. Ce que je disais il y a 15 jours, au cours de la réunion organisée par la Commission avec M. Dalli, j'ai rencontré ensuite la responsable du secteur OGM de la Commission Sanco. Je lui ai dit, ce que vous êtes en train de faire pour les États, c'est quelque chose qui doit être débattu aussi au niveau européen et donc au niveau de l'EFSA. Parce que je pense que les différents éléments qui ont été apportés sur les risques, en gros la présence des OGM dans les autres produits, la question économique et sociale, ce sont des notions, les effets sur les politiques culturelles, agronomiques, tout ça sont des nouvelles façons d'évaluer des risques qui ne sont pas simplement les risques scientifiques au sens classique qu'ils étaient posés au départ. Donc on a aujourd'hui un élargissement de la notion du risque par rapport à des pratiques agricoles, par rapport à des écosystèmes. On ne va pas rentrer dans les détails, mais on a aujourd'hui cette évaluation. Je pense que, et c'est vraiment, ça fait partie pour moi de la réflexion, il faut qu'au niveau de l'EFSA, je pense que vous serez d'accord là-dessus, qu'on soit en capacité de mener la recherche et l'évaluation sur l'ensemble de ce panel, parce que pour moi, ça fait partie aujourd'hui des questions clés. Et je crois qu'il est dangereux de renvoyer simplement aux États la possibilité de dire ce sont des risques que nous ne voulons pas, mais nous ne sommes pas obligés d'aller jusqu'au bout d'une étude pour pouvoir déterminer la réalité de ces risques. Et c'est pour ça que je suis très méfiant aujourd'hui sur l'approche qui est faite par la Commission pour dire on va permettre aux États, éventuellement même, de prendre des décisions pour des questions éthiques. Je pense que ce n'est pas un bon signe, parce qu'on ne peut pas s'opposer, je crois, de cette manière-là. Je crois qu'il faut que l'ensemble des éléments puissent être évalués. Je suis favorable à ce qu'on aille jusqu'au bout de la logique d'évaluation, mais de la manière la plus large possible, et qu'on considère les facteurs socio-économiques, environnementaux, etc., de la même manière que les éléments scientifiques de biologie moléculaire, par exemple. Donc je crois qu'on est sur une nouvelle évolution, et que si on abandonne de plus en plus la question de l'équivalence en substance pour une approche globale, diversifiée des risques, eh bien je pense que là, il y a un travail important qui aura été fait et que ça va nous permettre d'avancer collectivement.
Merci beaucoup. I have a final request for the floor from Angelica, and then I would like to close this morning's session, or hand over to Rita, in fact. Angelica, please.
Thank you very much, Helmut. My name is Angelica Hilbeck. I'm the chairperson of ENSA. I have a few remarks and also a response or a further question to Joe Perry, because your intention of clarification left me a bit more confused than before. You were saying that the comparative safety assessment is not a decision-making step prior to the risk assessment. Well, then maybe it just sits in the graph that you were offering at the wrong place, and maybe the terming is then a bit unfortunate, because a safety assessment has a pretty deciding value to it, or it has a decisive connotation to it. And you were saying that all it is that some data from molecular characterization is coming in. This is where you base your safety assessment on. So I don't think you can base a scientifically sound safety assessment just on a few data or some data from molecular characterization. So maybe just the wording is unfortunate and needs to be reworded and not made a safety assessment, because that obviously coming prior to a risk assessment is a bit illogic, at least to me and my colleagues. But that can be solved, I think, and from the discussion I take it here that most of it is about really defining the boundaries. We do all agree and acknowledge natural variation that is occurring. It is just that we need to define, it's not only about the quantity of the variation, it is about the quality, and which scientific criteria do we base our decision on, from when on a certain qualitative and quantitative difference is something that is outside of a boundary and needs to go into a risk assessment, and from when on is it inside of a boundary. So much of the discussion that I see is about defining clear criteria, cut-off points. What are the environmental harms that you include? Which are the ones you are excluding, so that everybody is very clear? None of us, just to make this very clear, none of us is intending to test everything. all of our initiatives and our engagement is towards going, how can we reduce it in a scientifically sound manner, be still inclusive enough, and exclude only those factors that are not meaningful, and how can we find those criteria and those defining moments. This is what the whole discussion is about, and that's where we have controversial meanings, and we need to come to a compromise, I think.
Okay. Thank you very much. I said in the beginning that I would be mindful of the timing, and we are a little bit running behind schedule, so I have the following suggestion. I think we will have a good opportunity to revisit the topics which we have now started to discuss in the subsequent parts of this workshop. And I would kindly ask you to come back to these points when we discuss the comparative document in its various parts. I would like to say at this stage that, well, first of all, thank you very much for MEP José Bové for his introduction and his contributions, to all the speakers of this morning and to your contributions. It's not the time to summarize yet, but I think we had a very good and rich debate. In fact, I'm very pleased that we managed very well to focus on the issues of substance here, and I have the feeling that we paved the way very well for the next part of this workshop, where we will enter into a more detailed discussion on the comparative document. So from my point of view, this was very good, and I would like to thank you for that. I now hand over to Rita. Thank you.
Thank you, Helmut. Thank you for all the speakers and the questions and discussions. I'm sure we have good discussion after coffee break, but now we have time for coffee. Since it's a bit late for the timetable, I suggest we come back at 10 to 12. But please be in time because we continue the webcasting, and it is important for audience outside this room that they can also follow us. So 10 to 12, please be back in your seats. Coffee is outside the room. Good. Welcome back to our meeting on EFSA stakeholder platform to discuss about EFSA comparative risk assessment and the stakeholder event. I give floor back to Helmut, our moderator. Please, the floor is yours.
Thank you very much, Rita, and also welcome back from my side. I hope you enjoyed the coffee break and had good and interesting conversations during that time as well. We are now going to continue with our workshop. After a very important and interesting setting the scene session before coffee, we are now moving to the second part of this workshop, which is about exchanging views on the draft EFSA guidance on the comparators. And we will set the scene for this discussion before the lunch break with two presentations first, and then again an opportunity for you to intervene, an opportunity for discussion. So I would now like first to invite Professor Howard Davies, who is Director of Science at the Scottish Crop Research Institute. I've been told that it will have a different name soon, but tomorrow. But today it's still the Scottish Crop Research Institute. Howard is the chair of the EFSA Comparators Working Group, EFSA GMO panel member, and he will give a presentation of the draft comparator opinion. So please, Howard, the floor is yours.
Thank you, Helmut, and I hope when I get home tomorrow I still have a job and somebody hasn't moved me out of my office. You never know these days. As Helmut says, my job this morning is to set the scene for the next few speakers in terms of progress with the comparators document and kind of the issues that we've been trying to deal with. And we've heard this morning in the way in which EFSA carries out its risk assessments, the starting point is to look at differences between the GM and its appropriate comparators, looking for intended or unintended effects of the insertional events. And to do that, clearly, as you're fully aware, we use a range of approaches from molecular characterization of the modification through to detailed compositional, phenotypic, and agronomic analysis, and of course also, where relevant, to look at the potential adverse effects on the environment. And all of these different parameters that we include in the risk assessment will differ in their requirement for comparators, and Joe Perry will be dealing with that in the next presentation. The important principle, I think, to recognize is that two tests are used by EFSA in the risk assessment process. One is the difference test, And again, you've heard a little bit about that this morning, to look at specific differences that may occur between the GM and its appropriate comparator. The conventional counterpart, as is also known, but also the equivalence test, which is to assess whether those differences in performance or composition actually fall within the natural variation, and that natural variation is assessed from a set of non-GM reference varieties. That terminology is quite important today, a set of non-GM reference varieties with a history of safe use. In fact, there were quite a few comments on the potential use of GM reference varieties in these particular equivalence tests. So the current EFSA position on the selection of comparators is to use non-GM lines, also termed the conventional counterpart, with a comparable genetic background for sexually propagated crops, and of course for vegetatively propagated crops, it's much easier to find an isogenic parental line to use as the comparator directly. But the extent to which the comparator actually relates to the GM event under assessment genetically depends upon the breeding pedigree used and also on the crop type used. And quite often there is variation in the closeness or relatedness of the conventional counterpart used with its GM for the comparison purposes. Clearly, for comparative analysis, particularly for stacks, we also require specific information from parental GM lines, which is important in terms of looking at potential interactions, for example, protein expression information. And of course, on a case-by-case basis, additional comparators will be required, for example, the use or non-use of specific herbicide treatments with regard to environmental risk assessment. Why are we actually carrying out this comparative analysis just now? Why are we looking at the guidance again? Well, more and more we're seeing that GM crops, GM plants are becoming increasingly complicated because of the various breeding schemes used to derive them, and the case in point being the complexity of stacked events these days. But also, with an eye to the future, we appreciate that there will be GM lines coming through which will be changed very significantly from the conventional counterparts, and that will provide a challenge. Again, we heard about that this morning. So I think the important thing is that we can evolve the basis on which we select comparators based on good scientific principles, which can keep pace with the technology developments, which are increasing at pace, while still fulfilling the regulatory requirements. And I think that's quite important. So the issues we've addressed in this particular document, which you've all read, at least the first draft, is to look at the selection of appropriate comparators for the risk assessment of both the single events and the stacks, to assess the potential use of alternative comparators such as negative segregants in the risk assessment process, also to look at appropriate comparators which may be used for the stacking of genes and traits, but not by conventional crossing, and clearly the selection of comparators in cases where the current comparative approach may not be suitable. And again, we heard about that this morning, and Harry Kuiper will be addressing that in the final session of this afternoon. Just to give you some examples of the complexity, we all know that once an event has been approved, that plant or that event can be back-crossed or integrated into a range of genetic backgrounds. Thus, 5 years down the line, the genetic background that event is in probably bears little relationship to the original genetic background. You also have the current commercial activity of third-party arrangements between companies where they're mixing and matching events, all coming from different breeding pedigrees, which clearly is a real challenge in terms of identifying one genetic comparator which suits all purposes. So that's, again, one of the reasons why we're actually looking at alternatives within this comparator document. On that particular slide, is the negative segregant, which could actually be derived from a final cross when you're making a hybrid, for example. And on a case-by-case basis, this may be more relevant in terms of a comparator than some of the other comparators which may be on the table for use. I've also mentioned the fact that new traits which will be coming on the market will be challenging in terms of finding an appropriate comparator. These would include, for example, GM crops with omega-3 oils enriched, and that's in the pipeline, or with amino acid compositions of proteins changed, or with modified levels of macro and micronutrients. A whole series of complex crops which could be developed over the next 5 to 10 years, which we need to be aware of in terms of developing our comparative analysis. I mentioned the use of the term negative segregant. What is a negative segregant? Well, it is derived from a breeding cross between two probably hemizygous individual lines, and these actual lines will lack the transgenic event or events in question. Okay? And as I say, they can be produced by self-fertilization of hemizygous GM plants, as an example. Right, okay, sorry, the camera was in the way, I couldn't just to remind, yeah, we are only thinking about allowing the use of negative segregants where they are derived from crosses between events which have already been risk assessed, okay? Not approved, this was one of the comments that came back through in the consultation, we don't use the word approved, hopefully in the next draft, but we would like to consider using the word assessed, okay? So what is the conventional counterpart, and what alternatives are we actually suggesting in this document? Well, first of all, it is stated in the document that the conventional counterpart, as we know and love it, is the preferred comparator. We're not running away from that concept. And there's also a statement in there that for the risk assessment of single events, we fully expect, in fact, there is no option here, we want to see the comparative analysis carried out with a conventional counterpart. That actually sets the baseline in terms of the risk assessment of stacks, where it may be very difficult to find or define a conventional counterpart as we define it. And bear in mind that the equivalence test, which I mentioned at the start, is always going to be there and will always include non-GM reference varieties anyway, so you will always have a comparison between non-GM materials in the field trials. And as I said, on a case-by-case basis, Joe may mention this, and Antoine, other comparators may be important in terms of delivering on the entire risk assessment process. In terms of the current definitions of comparators, you were exposed to this again this morning. The Regulation 1829/2003 states that a conventional counterpart is a similar food or feed produced without the help of genetic modification, and which has a well-established history of safe use. One could argue that new varieties coming on the market don't have a history of safe use, but that's not a debate I want to have today. Also, it's in line with the Codex Alimentarius definitions of a conventional counterpart, which is a related organism variety, again, in common use as a food, but that for the foreseeable future, these particular comparators will not be derived from biotechnology approaches. However, going back to actually the Codex Task Force report in 2001, there was certainly quite a few delegations which thought that the use of GM materials might be relevant as a comparator, whilst other delegations clearly did not feel that was relevant in terms of consumer acceptance as a comparator. And as I said in the previous slide, the statement that they come out with was that for the foreseeable future, foods derived from biotech could not be considered as meeting this criterion. That statement, I think, goes back to 2001, so the question I think we need to think about going into the future, not just now, but into the future, is what is the foreseeable future when, in fact, this is almost 10 years down the line? And are we entering a phase at some point in time where we will need to more fully accept the use of GM comparators within the risk assessment process? I also wanted to re-emphasize that the equivalence test always includes non-GM reference varieties with a history of safe use. So you will always have a comparator there which fits the definition of a conventional counterpart. The environmental risk assessment, and again that definition was shown this morning by a few people, and again it talks about the comparator being non-modifying organisms which are used in the comparative process for the ERA assessments. And I'm sure Joe and his colleagues will be mentioning this as the afternoon goes on. So to summarize, what we've tried to do in this document is to recognize the need to introduce flexibility in the selection of comparators based on what we believe to be sound biological principles, but also to align the selection of comparators with the evolving food, feed and environmental risk assessment guidance documents, also to comply with the definitions of comparators as currently present in the legislative documentation. And I'm sure, as I said, there will be a continued need to evolve the guidance based on further developments for the technology in coming years. So Rita mentioned the public consultation, and that's one of the reasons why we're here today, and I'm not going to repeat what Rita said, but basically from the written consultation and from your comments that will be trapped today, the plan is to take the draft opinion and to modify it appropriately following the consultation process before we move to adoption within the GMO panel. Thank you for your attention.
Thank you very much, Howard, for your very clear presentation. As before, I would suggest that we hear the second presentation in this set first before we enter into a discussion, unless there's an urgent question for clarification, which does not seem to be the case. So thank you very much, Howard. I kindly invite now Professor Joe Perry to the podium. Joe Perry is visiting professor of the University of Greenwich. He is member of the EFSA Comparator Working Group and, of course, also member of the EFSA GMO panel. And his presentation will follow up on Howard's presentation and will be about specific food and feed and molecular characterization and environmental needs for selection of comparators. Please, Joe, you have the floor.
Thank you. Good afternoon, everybody. Yes, I want to try and contrast molecular characterization, food, feed, and environmental risk assessment. They come from different fields: molecular biology, on the one hand, toxicology and ecology. Very different fields, so very different questions. I'd like to contrast them this morning, which I hope will explain why sometimes they require different approach and different comparators. In this very dense slide, at the top, for molecular characterization, food, feed, all I'm saying is repeating what everyone else has said, that our basis is that the comparator should have a well-established history of safe use. We define a conventional counterpart, maybe slightly different to that in Codex, in our document, but Codex is saying basically the same thing. And the idea here is that for everyone in this room, our ancestors have been pretty good at telling the difference between what food was safe and what food was not safe. Otherwise, we wouldn't be here. And that's been going on for about, well, at least 10,000 years since we've been farming. So there is the establishment over time of well-understood baselines for food safety. Scientifically, we will want to look at, for each endpoint, a range within which there is a history of safe use. Let's move now to ERA on the bottom row. You'll notice, first of all, that 2001-18, as has been pointed out, doesn't mention a history of safe use. The other thing I'd like to say here is that towards the bottom of the slide there, 2001-18, we've got information from releases of similar organisms that are not the parental conventional counterpart. Now, we've already discussed this morning. Let me try and clarify it again. I think there is no difference between my view and Dr. Bardosh's view. After talking to her, we realise that we both support, well, certainly to the extent that we both support the use of the conventional comparator, and that should be used in ERA and will be used 99% of cases. And there may be the odd case, the odd question, because ERA is so varied, and I'll come back to that later, there may be the odd question that requires a specific different comparator. But that will not be the norm, that will be the exception. Then, yes, OECD have spoken about familiarity, and they have said it's not synonymous with safety. I'd like to focus on the second sentence in that section. Familiarity with the environment comes from a knowledge of both managed agriculture and natural wild surroundings. My colleague, Professor Antoine Messiaen, will talk more about management and how important management is for ERA this afternoon. But for the time being, let's realise that management is vital. And in fact, in the words of Professor Chris Pollock, the sun's energy creates a zero-sum game, and if you want higher yields in agriculture, you are going to do that at the expense, usually, of biodiversity. And therefore, all agriculture has an adverse effect on the environment. And so we haven't got what we've got for food feed, which is a history of safe use. And not only that, but the problem is we have got few or no well-defined baselines, in the sense that adverse effects of conventional agriculture are dynamic. If you go back 60 years, we weren't using herbicides on a routine basis. So conventional agriculture changes and is dynamic through time. And using it as a comparator, we have to realise that that may create problems. Here's an example of one problem. We worked on the UK farm-scale evaluations of GMHT crops. 3 weeks before we were due to publish, when all the papers were in press, 3 weeks before that, the EU announced a ban on the herbicide atrazine, which is a key component of the weed control in conventional maize. The results showed adverse effects on wildlife for oilseed rape and sugar beet, but positive effects for fodder maize. Now, there was an immediate controversy there over whether the positive effects that had been found for maize would still be found had the conventional management not included atrazine. And indeed, ACRE referred to that in a statement they made. And what's the problem caused by? That problem was caused by a lack of a fixed objective baseline against which to compare GMHT. The conventional had changed, as it does. Pesticides, herbicides go in and out of the approved lists all the time. So, agricultural management regimes change temporarily, and the conventional comparators are dynamic. Now, let's focus now on field experimentation. The top row shows field experiments for food feed molecular characterization. Typically, these are our traditional agricultural experiments. Small plots, adequate replication. We can compare several treatments. As we will see, those treatments can include the commercial varieties that lead to the equivalence test. And there aren't that many types of field trial. There's compositional analysis, compositional trials, there's the agronomic. Often the data's got from the same field trial. We haven't got a big diversity. But Sebastian mentioned that there were the 6 issues that we look at in the ERA guidance document, and there were the 8 areas of concern that we look at, persistence and invasiveness being one example. So we've got a large number of combinations there which throw up questions, and these questions will be diverse. So there are many possible types of field trial for environmental risk assessment. But what are the other differences between that and food feed? Well, we demand realism in ecological experiments, and it is rare for ecological experiments to have such small field plots. Typically, they're much larger. But this means that because land is often restricted, our replication is restricted, and in particular, our ability to compare lots of different treatments is restricted, and so usually we're only comparing 2 or at most 3 treatments in a field trial. So we've got different approaches, and that leads to differences in our approach to equivalence testing. Equivalence testing being the way that we are proposing that we put any statistical differences that are found into a biological context and ask, is this difference biologically relevant? Is it important? And that's done thereby, as I said before, formalising and quantifying this nebulous concept of substantial equivalence. It's done through the equivalence test. Now, in food feed, this history of safe use, for any particular endpoint, we will have a number of varieties that are grown, and we can measure that endpoint, whatever that endpoint is, be it sodium, for example. We can measure it, and that will give us a range. And within that range, we know that that reflects varieties with a history of safe use. So that's how the equivalence limits are set for food feed testing. They are set by using conventional reference varieties in that experiment so that there are these additional comparators. For ERA, we don't have that history of safe use, we don't have the baselines, and so it's more challenging, I think. But we do have protection goals, and from the protection goals we can set limits of concern, and they indicate, that can indicate that a particular significant difference shows an adverse effect. And at lower tiers, those limits of concern will trigger further studies from lab up to field, and at higher tiers in the field, they should reflect actual harm. I should say at this point that we never use the GM as a sole comparator for ERA. We always have a non-GM comparator. The food feed equivalence test certainly provides an answer to millstone, as was mentioned by Dr. Mayer earlier. So this is how it is done. At the top we have the food feed context, and at the bottom the environmental context. We have a difference test, GMO versus comparator at the top. Then we also have an equivalence test to look at the effects of that possible difference, or to start to look at it in a formal way. This is not the end, but it's, if you like, the start of the analysis, this extra step. Then we can now forget about the near isogenic comparator. We look at our commercial varieties. They form a distribution, and from that distribution, the tails of the distribution are used to create the range within which we are looking at for safety, and our test then is against to see whether the GMO and its confidence in it limits fit within this range. And I may say that the equivalence test minimises consumer risk at the expense of producer risk, which is not the case for the difference test. So that's what happens for food feed. For environment, it's different. We have, again, a difference test between the near isogenic, usually, and the GMO. And that difference we can compare against zero to see whether there is a real difference. Then, we also have limits of concern for the difference. How big should that difference be, in our opinion, according to the protection goals, before this is deemed to cause significant harm and be an adverse environmental effect? And again, we're looking to see whether the GMO and its confidence limit comes completely within those limits of concern. So in both cases, we have an equivalence test. The equivalence limits are set differently for food feed than they are for ERA. And as Dr. Mayer cited the Perry et al. 2009 paper, that's where the concept is outlined, if anyone's interested in taking that further. And finally, just to say that obviously we're in a situation where we have updated food feed guidance adopted by EFSA in May 2008. The EC are still working on it. At some point in the future, it may transform into a draft legal document. We have updated ERA guidance adopted by EFSA in October 2010, and we have this current draft comparators guidance. It's important that we try to maintain coherence between these three documents. In the comparators document, you will see general text, then you will see text specific to MCFF, and then text specific to ERA. But two things to finish with that I need to stress, and they are that the ERA always uses the non-GM as a comparator. We don't have GM as a comparator at all. And in a great majority of cases, it will use a conventional counterpart, but it is case by case, and there may be a need to use an alternative comparator. Thank you very much.
Okay, thank you very much, Joe Perry, for your very clear presentation. With that, we have, with the two presentations which we just heard, we have been given a very good overview of the draft EFSA guidance on the comparators. And in the same way as before, I now invite for a discussion with respect to these two presentations. So it's your opportunity to raise comments. Please be brief and concise. We only have about 20 minutes for this part of the discussion before we break for lunch. In the afternoon, there will be a more detailed opportunity to go through the chapters of the draft guidance one by one. So again, please also identify yourself when you take the floor. I have a first request here, please.
Thank you. My name is Philip Knudder from Europa Bio. That's the European Association for Bioindustry. I was happy in the presentation of Professor Davis that he mentioned and emphasized the foreseeable future. Indeed, when we look back on the products that have been assessed before by EFSA, at least from a scientific point of view, you could also see them as rather simple products, and there will be increasing complexity, especially, as already mentioned, in the traits, but also the stacks. There will be a reality out there in the field, actually, that single events will no longer be commercialized. It will go in the direction of stacks and higher order stacks. Actually, I have two questions to the panel regarding also the changing environment. We also have a changing agricultural practice in, for example, in North and South America, where we have high adoption rates of above 90 or 95% of GM varieties in the field. And what does the term conventional actually mean in such a context? And the other part is that in case a comparator is difficult to identify, what is the experience of the panel members with the usefulness of omics approaches in the risk assessment? Thank you.
Thank you very much. Again, I would like to collect a few questions or comments before I hand over to the panel. So I think, Boet, you were next, please.
My name is Boet Glanhorf. I'm from the GMO office in the Netherlands, the National Institute of Public Health and the Environment. And I've actually a question for Joe Perry, and that concerns the limit of concern in the environmental risk assessment. Because what I wonder about, who is setting this limit? Is that the applicant who's doing the statistical analysis, or is that EFSA, or is that something for the risk manager? Thank you.
Thank you very much. Any further questions at this stage? Susanna?
Thank you. That's again a question for Professor Perry, comparing the GM with non-GM when there are significantly differences, and one wants to determine the biological significance of these differences. I still think, and I'm afraid I'm not alone with that view, that using 6 commercial varieties and using the full range for all 6 is not the right approach to see biological significantly differences. If you take the 6 commercial lines, take the means and the individual variation, and you use the widest one to justify a wider range, that's how far I would go with the comparator. And normally, if you have got 8 treatment groups, the one-way analysis is, in my mind, the right one. And I've drawn up a little graph I can show it to you later. Thank you.
Thank you very much. Any further comments? Angelika?
Not comments, but also asking again, do you have criteria? Are you working on criteria to define what is biologically relevant and what not? So I think the discussion about that, those criteria that define what is biologically relevant and what is not relevant needs to be led. We need to engage in that.
Okay, thank you very much. I think these were a couple of very clear, very precise questions. I think it's time now to hand over to Howard and Joe for possible answers on that before we open up again to the audience. I don't know who would like to start.
Yeah, deal with Philippe's questions. If I've missed one of your questions, please repeat. I would agree that if you look at the domination of GM soya, for example, it's going to become increasingly difficult to find non-GM appropriate conventional counterparts. But what I will say is that if there's a new event in soya, then clearly there has to be a non-GM parental line that you've transformed, so you will still have a conventional counterpart. If it's a stack, then clearly in our document we've given some flexibility options in terms of what you might use as a conventional counterpart in that particular case. With regard to omics technologies, I think this is potentially opening up a hornet's nest, because it's a little bit like targeted analysis. You still need databases which tell you what the natural variation is that you're actually comparing your GM crop against. So you still need to have this information. And I would say it's arguable whether some of these technologies actually suit themselves to a risk assessment process anyway. There may well be cases, for example, where you're changing endogenous pathways, where some type of targeted or semi-targeted metabolic profiling may be very useful in terms of looking at potential impacts on other pathways. But you have to be very careful about collecting what amounts to vast amounts of information in terms of how you translate that into safety analysis. And so I think be very, very careful. Did I miss your question, Sue?
Joe?
If I take Dr. Bardosh's comments first, I think we're in a situation where we would be unwise at the present time. Maybe in the future this will be different, but at the present time we can't trust the ILSI database. There is not sufficient environmental information from where these trials were done. That's why we insist that the commercial reference varieties should be planted simultaneously with the GM and the non-GM. Otherwise, I think we are in an unsafe situation, and I would worry that the limits would be too wide. Having said that, if we took just the range and not the 95% confidence intervals, we'd have an even wider range. So I think, again, we're being precautionary in taking the 95% confidence intervals. I appreciate your comment about the one-way ANOVA, which is often a very nice, simple way of looking at things and being brought up with it. I have immense sympathy, although I think that the statistical mixed model has such an ability to get more information, more refined information, that also has a real role to play. Regarding the limits of concern, this I think is a more difficult, it's a challenging question. I recall one of the most difficult things I think you can get biologists to do is to answer the question, what level of effect do you think causes harm, whether this is in toxicology or in environmental science? Very difficult question. Biologists don't want to answer you. We had this in the farm scale evaluations. We spent the whole morning trying to ask. We went round and round in circles. They just wouldn't give an answer. It's a difficult question to answer, isn't it? I mean, for a start, it's often very case-specific. We do give guidelines in the ERA. In the FSE, we used 50% change in abundance. There are guidelines in the ERA guidance document that talk about 30% and 20%, depending on whether you're working in the lab, etc., and depending on what the question is. This is a difficult one. However, I would throw the question back to you, and I would say, well, I guess I'm throwing it back to applicants. When applicants do a study, don't they have an idea of what size of effect they're looking for in that study? Surely you've got to, as a good scientist. This is just good science. And one of the reasons that we developed the ERA guidance document was that I think we found that there wasn't enough good science coming from the field trials that had been done previously. So, I think it's up to applicants to specify the level of effect that they're looking for in their environmental studies, be they lab studies or field studies, because, and the reason I mention applicants is because the applicant initiates the environmental risk assessment. But I do hope, and I do accept that this is the starting point of what may be a long process in developing limits of concern for different species in different circumstances. And I certainly hope that EFSA will have an input into that. Certainly we can comment on and will comment on the setting of the limits of concern by the applicant. And I also hope that as risk managers, the Commission will have a big input, and I look forward to the time when the Commission have a stronger body of expertise concerning ecology, and I know they're taking steps to address that. But that is important, that we all, risk managers, EFSA and applicants and NGOs, join the debate on setting the limits of concern. Thanks.
Okay, thank you very much. We have a possibility for another round of questions and comments from the floor. I'll start with Hartmut, then Pete, and a few more. Hartmut.
Yes, thank you. Hartmut Meyer. I just want to add a short remark on the limits of concerns, and I think this is exactly one of the crucial points, because this goes beyond a pure scientific reasoning. It's a normative issue in risk assessment, because in the end, you would not be able to find a scientific argument to say it's a 37% variation is correct. Beyond that is harm. It will be a decision based on whatever discussion and criteria. And the same goes with the problem formulation and to an extent also with the protection goals. This is a debate where you have to take decisions, normative elements of risk assessment. For me, this would include that a broader debate amongst a lot of stakeholders is done in this point, as you already have indicated. So anything that goes to an interaction between the applicant and EFSA to decide upon the limits of concerns, I think would not be really appropriate. So there needs to be a broader discussion on where to set them and to give more guidance on this process. So
Okay, thank you very much. Pete van der Meer.
Thank you, Chair. First of all, my compliments to the two speakers for very clear presentations, and I hope EFSA can have the PowerPoints on the website as soon as possible, if at all possible tonight, because there's another meeting on tomorrow where this will be very useful. Two specific questions about the comparators. First of all, has there been any discussion in your group how you would apply this model, which would work very well if you have a host and an insert or a donor, with fusion products beyond the taxonomic family? What are you going to compare this with? The second question would be, and this came up in a workshop recently, it links a little bit to what Hartmut raised, the concern, the limits of concern. This links actually to the problem formulation and what is biologically relevant. Suppose you have a case, insect-resistant maize. You're comparing it with a non-modified maize, and you have insect resistance there too. would the fact that in non-modified maize insect resistance would not raise the questions about, for example, resistance development, would that be a reason not to include it in your risk assessment? So is the fact that you include certain aspects, or you do not include certain aspects with non-modified maize, would that be a reason not to include it in your assessment of the modified maize?
Thank you. Beatrix.
Thank you very much. 2 points I would like to ask or to mention, because I was in the presentation about flexibility in the form of the comparators. So to my point of view, beside of other reasons, that should be based on scientific data and especially on monitoring data. So do you have any indication, recommendation in the context of that? Because until now, we do not have monitoring data, not in the food feed safety area and not in the environmental data, but so really hard scientific data. So that is one point. And the second point also refers to the limits of concern. And my question there is, as you said, Joe, that to a certain extent it will be up to the applicant. We have heard that it is some form of normative, some form of normative setting. But in any case, it is very important in the context of the field design to know what, for example, an applicant has taken into account, how he has designed his field and his field experiments in order to really know which sensitivity is in the test design. So do you have some thoughts about that or some recommendation in that context?
Thank you. And I take another question. I think here. Yes, please.
My name is Sławomir Sowa from Plant Breeding and Acclimatization Institute, National Research Institute in Radzików, Poland. I have a question to Professor Perry. You mentioned, and this is also in the draft guide of documents, that for the risk process comparison, both for the food and feed and environmental risk assessment, we need comparator that should be a food or feed produced without the help of genetic modification, and for which there is a well-established history of safe use. So my question is, what would be the comparator with a well-established history of safe use? If you could give some more details on that. Would it be a variety that is on the market for many years already? Or could it be a similar genotype for which a risk assessment was already performed? Or could it be a variety that is derived with the means of mutagenesis, for example, that is present on the market but not risk assessed? Thank you.
Okay, thank you very much. I think it's now time again to turn to Howard and Joe in order to answer some of the issues which have been raised.
Okay, if I understood your question, it's about a GM event that derives a product which takes you beyond the taxonomic boundaries. Is that what you were saying?
No, I'm referring to fusion products. So if I fuse just beyond the taxonomic family, for example, I fuse the cell of one species with another one, maize and wheat, for example. Okay? So you have fusion products. Those are included in the definition of GMOs. Your comparator here works fine if you have a host and an insert, and you compare with the non-modified host or parental organism. But if you fuse your cells from a large distance, how would your system of comparison, how would that work?
That's a very interesting question, because whilst fusion products, and you're quite right to bring it up, fall within, as far as I remember, the legislation, we've never actually seen in EFSA, in my time, nor even in the Scientific Committee on Plants in my old time, any example of a fusion product that's been brought to market. And it's actually not something we've even discussed in the comparators group, to be frank, Peter. I'm sure there is an answer, but I wouldn't want to give it off the top of my head. In terms of, there was a question about flexibility in the monitoring, and certainly with regard to the scope of this guidance document, monitoring does not really fall within this discussion today for me, because it's kind of it's almost a cross between risk assessment and risk management, and I think it does fall out with the scope of this particular document that we're discussing. in this session. Sure.
To address Hartmut's comment, yes, I think a broader debate is necessary. I agree with you. Don't forget, though, we have protection goals. So the risk managers have protection goals that are the basis, or should be the basis for these limits of concern. Of course, different member states have different protection goals, and that's a further complication. To address Pete's question, I think certainly within ERA we deal with insect resistance management, and that's including this section on target organisms. I hope that that's brief enough. Beatrix's question, sorry. As Howard said, it's without the scope, but I think the ERA guidance document does make mention of the fact that there is, in the future, expected to be some feedback between monitoring back to inform future ERAs, but I don't think I can go further than that. Regarding your other question, which I think is more serious, about the limits of concern, in the future, according to the ERA guidance document, applicants have got to specify for each study, whether it's a lab or a field study, the effect that they are doing that study to detect. That seems to need to be reasonable. They also will have to do a power analysis to give EFSA an indication of what the power of that study is to detect that effect. And in fact, that's all laid down in the ERA guidance document, and there are a set of 35 shall questions, or questions that relate to where the ERA guidance document says the applicant shall, which will in the future be checked at the completeness check stage that relate to the statistical section. This, I don't think, puts an unbearable burden on applicants. It's just good science. It won't take long, but it's important that we have that information, and I make no apologies for the fact that we demand it. Regarding the question here about what would a comparator be if it had a history of safe use, how do you define it? In general, it's a non-GM variety of the same crop which is appropriate for the receiving environment in which the field trial is being done, and again, that has to be justified explicitly why it is appropriate. It has to be appropriate. It's a variety that would usually be grown in that environment.
But it would not preclude a product which is being developed, for example, by traditional mutagenesis, if it was appropriate for that receiving environment.
No, I would say that mutagenesis has given rise to Golden Promise, the barley variety that is made in all UK beer, which is such a fantastic foodstuff that I can recommend it. And it's safe too.
Thank you. Talking about foodstuff, I think we are about to break for lunch right now, unless there are urgent requests for the floor at this stage. I think we jump into a very detailed discussion of the draft EFSA guidance on comparators, which is very good. We are going to continue with that in the afternoon, when we have the possibility to present and to discuss the various chapters of the draft guidance. So I think we are quite well on track. I would like to thank our two speakers at this session. I would like to thank you for your active contributions. Before I hand over to Teresa, and please just one more minute to close this morning's session, I would just have a small request to the 12 speakers of this afternoon, or 12 should be, please stay for a couple of minutes with me in the room. I would just like to have a short pre-meeting with you. So with that, thank you very much. Enjoy the lunch break. I'm looking forward to seeing you again. And I hand over to you,
Helmut. It's a morning session, and I would like to ask you to be back on precise 2 o'clock so we can continue in a timely manner, because we have many things to be discussed in the afternoon session, which are very interesting, so we want to keep all the time reserved for that case. So please have a nice lunch and be back at 2 o'clock here. Good afternoon and welcome back to EFSA event about selection of comparators for GM assessment plans. Happy to join the discussions for this section, and also I want to welcome all those back to our meeting who are following via webcasting. Over 500 people have joined us today outside this room, which is very good for discussion. So interesting events and interesting discussions will continue. I give floor back to Helmut, please.
Thank you very much, Rita, and also good afternoon from my side. Welcome back. I hope you had a very nice and refreshing lunch break. And I'm pleased that we are now going to continue with our workshop. The program for this afternoon is, after we have set the scene this morning and already entered into a discussion on the draft EFSA guidance on comparators, now to go into some more detail. We will have 4 brief sessions where 4 main chapters of the draft guidance will be introduced briefly. 2 sets of comments will be made, and then you have the possibility to provide questions and comments from your side as well. And then we change the subject. The 4 topics which we are going to discuss in this sequence will be risk assessment of single events, risk assessment of stacked events not from conventional crossing, then we deserve a coffee break, and we will finish with risk assessment when no comparator is available. And then with some concluding remarks, the day will be wrapped up. So this is the program for this afternoon. And without further ado, I would like to start the first round of introductions and discussion, risk assessment of single events. Here we will have 3, maximum 5 minutes interventions here from the podium. The comments received during the public consultation process will be provided by Professor Dr. Antoine Messiaen, who is a research manager at the National Institute for Agricultural Research in France. And then we will have 2 invited comments, one by Wolfgang Reichenbecher, who works with the Federal Agency for Nature Conservation in Bonn, Germany, and one by Dr. Peter Windels from Europa Bio, who is Bioscience Regulatory Affairs Department of Biocrop Science. So in this sequence, and each of you has 5 minutes, I would like to give now the floor to Antoine, please.
Thanks, Helmut. I've been asked to summarize the public comments received on this section. And but first of all, I'd like to come back to a point we have been discussed this morning with some comments, and Joe Perry mentioned the importance of management systems. And I'd like to highlight 2 aspects. First, that as any new techniques in agriculture, so the introduction of GM may induce indirect change in the management systems, and for example, the change of herbicide regime for herbicide tolerance. But beyond these direct changes of practices, you may have further changes in the practices for soil tillage, for example, or in the crop sequence. And so, of course, this aspect is not specific to GM, but anyway, it should be considered in the environmental risk assessment. And the second aspect I'd like to highlight is that any differences we may find in the different tests when comparing the GM plant and the conventional counterpart, which might be detected should be put in place in the context of the receiving environment and the management system. And so it's not easy to do that, because we have a great diversity, and I quote here the part of this recent, the updated environmental risk assessment guidance document, in which the EFSA, the panel, acknowledges the fact the diversity of the current management systems, and systems from the very intensive to organic, and with a wide range of systems in between. And that the environment risk assessment should compare a range of GM-based systems to a wide range of conventional systems. So that makes the choice of comparator a little bit difficult. And so we have set some principles and preliminary approaches, but any idea would be useful to help us implementing this approach. As for the single events, so I will be short, because in the background, because nothing new, the current principles are unchanged. The risk assessment of single events always include a conventional counterpart. And then in the background, what we have taken into account is the fact that we have different sources of information, and Joe already mentioned this aspect, so I won't detail that. And but I just want to point out that as for the comparative field studies, field trials, you've got a wide range of field trials, and I would distinguish this so-called plant-oriented. When you are looking for assessing potential unintended effects, you will be looking at very carefully the differences between GM plants and the conventional counterpart. And in this case, of course, these two genetic backgrounds the genotype should be as close as possible to avoid any confusing effect. But when we are looking at, well, I mentioned the importance of management system, we also have the field trials system-oriented, when we are looking at the whole picture, taking into account all the aspects that may change in the management systems. In this case, the focus will rather be on the management techniques. So that should be taken into account, and that's why the panel and working group has discussed the possible use of additional comparators and of additional management regimes. We have received 16 comments on this section, and some of them were referring in fact to the risk assessment strategy, and in fact it was not very well, it was not the purpose of this document. And so we are considering cleaning the document to avoid any confusion and to cross-refer to the appropriate guidance document, food and feed or environmental assessment. And most of the comments were on clarification and guidance, but we will listen to more comments, I'm pretty sure. And so I have summarized this received comment in 4 categories. First, the use of GM commercial varieties as reference. So this has been addressed this morning, and so it's not considered by EFSA for the time being. Howard discussed that this morning. Second aspect, it's about the negative segregant, which is not a non-GM comparator because it would never be able to discard any unintended effects with such negative segregant. And we will make it clear in the document. And you may remember that the definitions of negative segregants were in that section. Of course, it will be moved elsewhere because anyway, single events are not using as a sole comparator negative segregants. There may be additional comparator. The third comment is about the potential effect on back crossing. So as far as it is possible, of course, we should use the near isogenic, but it is not always possible. For example, if we want to assess the differences into various receiving environment and climate, you will have to use varieties adapted to the local environment. And to do that, if you want the applicants to perform various field trials under different conditions, you need to have different varieties. And to produce this variety, you need back crosses. So, but at the end of the day, we request that these back crosses be justified and as limited as possible. And the final comment is about a request to EFSA to better specify the criteria for selecting alternative comparators. This has been addressed this morning, and in fact, I would like to mention that we will make it clear that in the great majority of the case, anyway, we will have a conventional counterpart anyway. And Joe commented one example with dot tolerance this morning, so that only in some very specific ergonomic threat and field trials, and we will make it clear. Thank you for your attention.
Thank you very much, Antoine. I would now immediately like to invite Wolfgang for the first invited comment to this topic, risk assessment of single events. Wolfgang, you have the floor, please.
Thank you, Helmut, and I would like to thank the organizers for inviting me and for giving me the opportunity to present our comments. Comments presented here and the ones we've submitted in writing earlier this year are a joint effort of the division I'm working in, the division GMO Regulation, Biosafety of Federal Agency for Nature Conservation. I'd like to show you an overview about our comments. Some of them are related to Chapter 3.1, most of them actually, and I'm going to explain them in more detail through the next slides. And two of them are related to other sections which are covered in the afternoon later. The first one is in the background chapter, the chapter that is right at the beginning of the draft document, the legal references need some completion. The second main comment is that negative segregants are useful as additional comparators only, and I'm going to explain that, and we think that the impacts are not sufficiently discussed throughout the draft. The third main comment is that the use of GM lines is, as only comparators, is not in line with the Directive 2001. And all these three comments relate to Chapter 3.1. And the next comment is related to another chapter. So we think that the general approach to risk assessment of stack events as presented in the draft does not fully comply with the way it is presented in the EFSA guidance document for stack events, so that there is a difference, and would like to point EFSA towards this point. The next point has already been mentioned a couple of times this morning. We think that the comparative approach is not suited in two cases. And the one case is that the conventional counterpart cannot be cultivated in the same environment. For example, when the GMO is resistant to abiotic stress. And the second is when the GMO has got major changes in metabolism and characteristics, and that is because there is no comparator. So in these two cases, comparative approach is not suited, I think. And the last point I'm going to explain in one of the slides. So those 2 points in grey are not related to Chapter 3.1. And now about the background as provided by EFSA. The background stresses the central role of selection of appropriate comparators for risk assessment, and we agree with that. However, the legal reference mentioned there is only to Regulation 1829. There is another reference that is to Codex Alimentarius, but that, as far as I understood it, is not a legal reference, at least not in the way it is for the regulation and for Europe. What we would like to see is that those legal references mentioned in the background, which is the starting chapter for the draft, is completed by references to Annex 2 of the Direction 2001, which is mentioned throughout the draft, but which isn't mentioned in the background chapter. And also, it should be completed by reference to the decision 2001-2, which is giving further guidance to how to apply the requirements laid down in Annex 2. And as has already been mentioned in the morning, Annex 2 is saying that characteristics of the GMO should be compared to the non-modified organism, so meaning the recipient. On the next slide is about negative segregants, and we've seen that there is in chapter 3.1 there is a paragraph about 8 to 10 lines with a description about the characteristics and use of negative segregants, and we appreciate that that is included in this chapter. And also using negative segregants as additional comparators is welcome. However, what we miss not just in this chapter but also throughout the whole draft is a comprehensive discussion reflecting on 3 points, and one is the potential and actual contribution of backshotting to eliminate unintended effects derived from genetic transformation. And second is to reflect on the approach to detect unintended effects via comparative assessment only. And then, as a sum, to reflect on the chance for unintended effects to remain undetected throughout risk assessment. And because this comprehensive discussion is missing, we think that the use of negative segregants as only comparative is not sufficiently scientifically justified. So they should always be used as additional comparators. And besides, it's not in line with the Annex 2, and therefore it's quite important to mention that Annex right in the background in the starting chapter. There is one issue which is occurring in chapter 3.1, which is about additional treatment, but also in other chapters throughout the whole draft, and that is about the treatment management aspect. And I've quoted lines 339 to 3.42, and our impression was that here the draft already starts to specify some requirements for additional treatment. And our impression was that this should not be covered in this guidance document, but to avoid conflicting guidance, it should be better covered in main EFSA guidance document. So it's not actually covered by terms of reference and not related to selection of comparators. That is another point we'd like EFSA to consider. On the last slide, I would like to mention 2 additional comments. One is related to comparators, and the other one is related to comparison in general. So, as I've mentioned with the background chapter, there is an emphasis on food references, and environmental references such as the directive and the decision are not equally balanced. It's the same with when comparative approach is presented throughout the document. Then the draft relates mainly to food aspects and does not reflect sufficiently to environmental aspects. And another point, which is for comparison in general, what we've seen in recent applications and in studies provided with applications is that the starting material for field trials regarding expression, composition analysis, agronomic studies is most checked for identity, but it isn't always studied for purity. So sometimes it isn't checked, and when it's checked, it's not always demonstrated that the starting material is devoid of any other transgenic traits. And we think that is increasingly important because companies tend to stack events, and what they do is they plant a different event next to each other within their field trials, and so there's an increased chance for commingling, commixing, and contamination. Thank you for your attention.
Thank you very much, Wolfram, for this comment, and we immediately move on with Peter Windels from Eurobio. Please, Peter, you have the floor.
So first of all, I would like to start with thanking EFSA for inviting us here and also for recognizing the complexity of this issue we are discussing today. The slides that I will present this afternoon, you will see basically they are some highlight comments that we also used or that we also submitted in the frame of the public consultation. And also, I would like to know that some of the comments we have in the slides have already been discussed this morning, so I certainly will stick within my 5 minutes. Okay. So first of all, we had some general comments, and the first general comment we had is on consistency of EFSA guidance. Because it's really important for applicants that we have a consistent guidance to which we have to build our applications. And we know that in the past there have been some updates on EFSA guidance. There has been the update on the food feed risk assessment, also the environment risk assessment. And also now in this document on comparators, or the draft document on comparators, there are topics that also touch on issues that have been discussed in the food feed and in the environmental risk assessment guidance. So therefore what we would like to see is that in the end, ideally, one guidance document that captures everything. So basically it relates to the purpose and use of new guidance in light of the previous guidance. It's also, the new guidance should always be complementary to existing guidance. And also for applicants, it's really important to have a clarification on the status of the older guidance. The second comment the second general comment is on clarity on requirements. And I will give one example, but my colleague, when discussing about the requirements for stacks, will give some additional examples. But for instance, we know that the draft document makes a distinction between, on the one hand, molecular characterization and food feed risk assessment, and then the environment risk assessment. It would be important for us to know which studies fit under molecular characterization and food feed, and which studies fit under environment risk assessment. Because there are differences in requirements, and we would like to see that clarity in the final comparator document. So these were the two general comments. So now I also have two additional slides on more, I would say, technical scientific comments. The first one is on the use of reference varieties. And what industry has seen in the draft document was that there is a discrepancy between the definition of reference varieties in the document. On the one hand, sometimes it refers to a set of non-GM reference varieties with a history of safe use. But then also reference varieties are defined as representative varieties that would be normally grown in the areas where the field trials are performed. So we would need to have clarity on that, and certainly from our side, the proposal would be to define the reference range as a range which includes basically commercially available GM crops, so which are basically approved and safety affirmed, together with also non-GM reference varieties. Because in the end, certainly for import dossiers, including GM varieties in your reference range would really reflect the reality of the current agricultural practice in the regions where these crops are grown. And for that, I just want to highlight one slide which basically shows the percentage of GM crops in specific crops, and also in basically it's a global overview. You see, for instance, that in the case of soybean, 81% of the soybean acreage is GM, which in the end, if we as applicants can include GM varieties in the reference range, would really reflect the current agricultural practice. One additional slide, and one last slide basically, is a slide on protein expression analysis. And here the comment we had is that our understanding is that protein expression analysis is carried out to determine exposure to the newly expressed proteins. So basically, protein expression analysis is not a comparative assessment. So therefore, that's also why for protein expression analysis, we do not have the exact same field design as for the composition and agronomic assessment. But one additional topic that we would like to highlight is we have seen in the draft guidance that reference is made to the fact that in the case of protein expression analysis, you also need to include a comparator, because that comparator can be used to validate the specificity of your assay that is used. Our comment is, in fact, why would you need to have a comparator in the case of protein expression analysis if the specificity of your assay has been determined elsewhere? So therefore, we do not really see the need to have a comparator also in that case. So I think these are the slides that I have prepared, and these are the main comments we have. So thank you.
Okay, thank you to all the three of you, Antoine, Wolfram, and Peter, for your very clear presentations. We have a few minutes left for discussion on this particular matter now, risk assessment of single events, choice of appropriate comparators. Who would like to start? Susanna and then Inge, please.
Thank you very much. EFSA cannot but recommend it for trying to find the right comparator. However, I cannot see any problem for finding the right comparator, non-GM conventional comparator, for any GMO targeted the change in composition. For food and feed experiment, it would be very easy to add chemically the altered compound to the control line. For instance, if you have got an increased omega-3 content, you can add the same amount of omega-3 to the control and carry out the experiments for food and feed, for toxicology, in case of environmental experiments, and artificial diet is prepared for target or non-target organisms. Thank you.
Thank you. Again, I'm collecting a couple of questions, so Inge, please.
I just want to make a comment on this story that there might be problems with products coming from a field trial. If you have stacked events, what was the scenario mentioned there? We should keep in mind that the products of field trials are never commercialized. So if you have stacking of genes in a field trial, there's no problem at all, except maybe for the researcher who might have problems with the results. But it never is commercialized and will never come into the market or somewhere else in the environment.
Okay, thank you. Any further comments or questions? Adinda?
Thank you, Helmut. So first off, my name is Adinda de Schrijver, and I work for one of the risk assessment bodies in Belgium. I have actually one question for clarification, and it's on the terms. It's on the term, and I quote here, a non-GM variety with economic properties similar to the GM plants, and the term reference variety. So we understood that the first term is used in the context of environmental risk assessment, and the latter one, reference variety, in the context of food feed risk assessment. And we wondered if these terms are identical and refer to the same type of comparator. So I'm asking this question because we had some questions from people from the council, if why no reference varieties are used in the ERA. So I would like to have some feedback on this issue. Thank you.
Okay, thank you. I think there were three very clear questions, and I would like to turn to the podium, maybe to answer the one or the other, and then we can see if there are further comments from the floor. Is there anyone from the podium who would like to start? Claudia.
Okay, I'm Claudia Paoletti from EFSA. Regarding the first question, the one with the reference to the legislative framework, the comment is valid. In the version that went for public consultation, we were focusing mainly on the requirement of 1829, but indeed in the version that will be revised, we will also make reference to 2018 and the Commission decision of 2002-623. So that point is well taken. I think I can pass the floor to Antoine for the issue related to the ERA and the treatment management.
Yes, we have quite a lot of questions. We'll try to make it quick. But first, I take your points. That's why I'm insist on the fact that it should be as close as possible, and that in any case we should justify the breeding scheme, and so EFSA is looking at this aspect. As for the additional treatment, we should not be part of this guidance document. I tend to disagree. Well, as I mentioned before, for this system-oriented, when we are looking at the whole picture, and so the effect of the related management techniques to the GM plant, anyway, you should take care of the way the GM plants are grown, and not just to stick to the GM plant independently of the management system, production system, receiving environment in which it would be put into the market. So I think that this system-oriented approach is taken on board some of the comments we had this morning. And including for plant-oriented field trials, we still have to take into consideration potential interaction between the GM plant, the behavior plant, with this additional component. For example, this herbicide regime for herbicide-tolerant crop. Which study in the environmental risk assessment and versus food and feed, I take the point also that so far the compositional analysis is mostly driven by the food and feed requirements, but the environmental working group has been looking at the design of the requirements that may end up with some additional clarifications on the way the environmental risk assessment uses this data. Maybe, well, I won't comment the questions on the adding chemical, because I think that will be addressed later today. And clarifications are in there about, well, for sure the text was confusing. And so reference variety anyway is for food and feed only. And as for the agronomic, well, the variety as with agronomic property as close as possible, it's only in the case where we have some specific field trials and specific agronomic traits, like Joe mentioned this morning, this drought-tolerant example. And so that will be clarified.
Okay, thank you very much. Any further questions to this topic? Yes, please.
Specific clarification. We are talking about the comparators here. It's fine for the feed and food. We can use the whole natural variability of the species, but for environmental risk assessment, it's much more difficult. We are talking about the type of the comparators, but perhaps, at least from my point of view, it would be good for the risk assessors and even for companies to know how to arrange the trials, how many repetitions and set up the acceptance criteria for the comparison, if it's intended in material or whether you would like to deal with that.
Okay. Thank you for that question. I'm looking if there are any further and final comments to this part of our discussion before I give the floor to the podium, and then we move to the next subject. So if there is no request for the floor at this stage, then I would kindly ask Antoine for the and then we...
Again, there is no non-GM commercial reference varieties for the environmental risk assessment. We're using the limits of concept that was presented this morning, and we will always have a conventional counterpart in there. And I think as for this design of field experiment, we have a specific section in both guidance documents, and yeah, so I think it's in there. And if it's not clear enough, I think please advise the EFSA to any clarification. But the recommendations and guidance are in this document anyway.
Okay. Claudia?
I can just add to what Antoine just said, is that in addition of having a very detailed description of the field trials in the ERA guidance and in the new to come food feed guidance, there is also the already adopted opinion in 2010 by the GMO panel on the statistical risk assessment, where we have a full section of experimental design requirements where different cases are covered. So there is a lot of details there that can integrate what's already present in the respective guidance documents.
Thank you, Claudia, for this important clarification. I think that was a very good discussion on the first set of topics this afternoon, single events. I would like to thank our three speakers. Thanks for your contributions. We are now moving on, which means a change here on the podium. I would kindly ask our three next speakers on the risk assessment of stacked events to come up to the podium. That is Professor Howard Davis, Annick Clézier, and Dr. Andreas Heisenberger. So we are moving to the next subject, the next chapter, so to say, of the draft guidance on comparators. Risk assessment of stacked events now is the topic, so we're moving on from single events to stacked events. Professor Howard Davis will briefly summarize the comments which have been received during the public consultation on that part of the guidance, and then we will hear two invited comments, one by Annick Clézier, who is working with Europa Bio, and another one by Dr. Andreas Heisenberger, who is Deputy Head of Unit Land Use and Biosafety at the Environment Agency in Austria. So, Howard, you have the floor, please.
Thank you. Well, my job is made a little bit easier because I think a lot of the topics were discussed or touched upon during Antoine's presentation. First of all, clearly there were editorial clarification issues that we needed to deal with in this section, and they will be dealt with accordingly. There were a number of comments that maybe the entire document, not just this section, was delving too much into the risk assessment side of things. That comment came from quite a few of you guys, and that again I'm sure will be taken on board as we revise the document. So we will try to focus primarily on the selection of comparators and the justification for the selection. And in fact the risk assessments, as I've already heard, will be dealt with specifically in the new guidance documents, which are on their way over the next few months, we hope. So, again, some of these comments have already been touched upon by previous speakers. The issue of the use of the negative segregant as the sole comparator was questioned, particularly in the light of definitions within 2001-18, which a previous speaker alluded to. Applicants were also questioning how they could convincingly demonstrate that a conventional counterpart is not available. Also, as you heard in the last presentation, some groups were keen on the idea that GM varieties should be allowed as reference varieties within the equivalence testing trial. Also, comments about protein analysis being more about exposure rather than a comparative assessment. And there were also comments on the nature of the herbicide treatments that were used in the comparative studies. So these are kind of generic issues that came through quite a few of your comments. And that's where I'm going to leave it, because I think it's more important to maybe discuss them, to go over the same points time and time again, because they relate to the singles and the stacks. Thank you.
Thank you, Howard. I would now like to invite Annick Clézier from Europa Bio to give her invited comment, please.
Okay. I would like to thank EFSA in turn also for inviting Europa Bio to present comments on the risk assessment of stacked events in the context of this comparator document. I'm afraid that I will be repeating some of the comments that were already made, and in fact some of the comments that we've made are already addressed, but I will go over quickly for discussion. So first of all, I would like to present some general comments as my colleague did in the context of the section on stacked events. One comment is related to the need for clarity for applicants to be able to present applications according to the requirements. I just put down one example that was also mentioned by Howard, where I quote the guidance: applicants can convincingly demonstrate that a conventional counterpart for the stack cannot be made available. We asked the question, what would constitute convincing demonstration? Another example, another general comment is on choice of terminology, and again that was touched upon by Howard this morning. Europa Bio commented that the condition for using negative segregants in the comparative assessment of stacks should be the completed risk assessment and not of the corresponding GM events and not the approval. The GM event approval does not provide more reassurance with regard to safety than the scientific risk assessment itself, whilst there might be a significant delay between the EFSA scientific opinion and the approval. A few more specific comments. One related to the additional comparisons. The comment that we made was that the scientific rationale for having both the treated and untreated sample analysis for comparative assessment for composition and agronomic phenotypic studies for herbicides or on singles is unclear to us. This requirement is still part of the draft updated guidance for the risk assessment of genetically modified plants for and derived food and feed. But our proposal is that in absence of that scientific basis for the requirement, further complexity in the field trial design should be avoided for stacks. In other words, we recommend that a clear distinction is made between singles and stacks with regards the need for both treated and untreated sample analysis and comparisons. And the last comment relates to the assessment of subcombinations of events present in the stack. Here we just reiterated comments that we made earlier, namely if behavior has been characterized and confirmed in a single event as safe and absence of antagonistic and synergistic interactions has been established, then combining transgenes into stack is not expected to produce any greater risk of unexpected novel genetic combinations than combining the tens of thousands endogenous genes in conventionally bred crops. And in that regard, and the general approach that was presented, stack approach that was presented, namely the single being the prerequisite, single risk assessment being the prerequisite of the stack risk assessment, and then the subcombinations to be covered as independent stacks, we requested further clarification on the implications of that proposed stack approach. On the one hand, for the risk assessment, meaning particularly very clear terminology is needed. We've pointed out, I think, some examples on terminology like covering or considering the subcombinations when distinguishing between the environmental risk assessment and the import requirements, but also generally if no synergistic or antagonistic interactions was demonstrated on the highest order stack, clarification on what is exactly needed in terms of data requirements when reducing the number of events. And then on the other hand, also the implications on the approvals itself, and there the comment is more related to, okay, how will this turn into an approval decision? For example, the differentiation between a commercial product, a commercial stack product, and the subcombinations as a segregating progeny. An example of the questions we have there could be, what about the consequences for detection methods of these subcombinations and the related costs? That's it. Thank you.
Thank you very much, Annick. And we move on with Andreas from the Environment Agency Austria for his invited comments. Andreas, please.
Okay, yeah. Before I start, I also want to thank EFSA for giving me the opportunity to present the Austrian comments and the Austrian point of view with regards to stacked events and the guidance for the choice of comparators. So let me start that we are fully aware and we acknowledge that it's much more difficult to find the suitable comparators for stacked events than for single events. But although we are aware of that, we also believe that some certain simple criteria should be fulfilled, and we believe that this is not completely reflected in the document. So let's start with the two main points, or I want to point out the two main points we had. The one is the non-GM comparators and the other one, the negative segregants. Both topics have been discussed already, but I want to give some reasoning why we think that this needs to address a little bit better in the document. So let's start with the conventional counterpart. Of course, we are aware that the non-GM parental line is not always available, but the document opens up the choice of comparators to use negative segregants, for example, or lower stacks, single events, in case no GM counterpart is available. And I think this is one of the few cases where we are completely in line with Europa Bio, because we say we need justification, we need more guidance on how this justification can be done. I think this is not only important for the applicant, but also for us, because we need to judge if this justification that no GM or no conventional counterpart is available, on which criteria this is made. The document, as I said, open up to use negative segregants and GM comparators, actually, if we talk about single events or lower stacks, if no GM counterpart is available. We believe that in any case, we need an appropriate non-GM counterpart. This has been pointed out by my German colleague before for the single events. This is in the legislation. There is no way around it, in our point of view. And if there is no parental line available, which might be the case, we acknowledge that, we need some as close as possible comparator. And if you look at the Codex Alimentarius, the document of 2003, it's written also in there, as close as possible. And we believe that this could be a suitable model also for the comparator document we're discussing today, if a justification is given why this is done and how this as close as possible comparator is chosen. The next point I want to make, and that's a quite important point to us, is the use of negative segregants as comparators. The document also states if no conventional counterpart is available, then negative segregants can be used as sole comparators. We believe that this is not in line, first of all, with the legislation. If you look at the 2001-18, we have heard already that a non-GM counterpart should be used. On the other hand, a negative segregant has been modified at some point in the breeding history. And if you look at the 2001-18, it's a GMO. So these two don't go together in our point of view. So negative segregants, in our point of view, fall under that definition and should not be used as non-GM counterparts. The other reason is a more scientific one, and that can be found in the document, in the comparators document. Because if you look at the single events chapter, you can read there that effects of the genetic modification process cannot be completely discounted using a negative segregant as a comparator. So, first of all, this acknowledges that the negative segregant is a GMO. And second, therefore, the possibility of unintended effects in single events must be assessed using a non-GM conventional counterpart. So we believe if this holds true, and this has been stated by the EFSA panel, for single events, this also holds true for stacked events. So just one last slide to sum it up. We think that to assess stacked events in a comparative way, we need a set of different comparators. The most important one, we need a true conventional counterpart, if available the non-GM parental line, or a conventional counterpart with the same or a similar breeding history. And if no conventional counterpart is available, we need a very clear guidance on how this should be justified. We need a set of criteria on how this could be made and how this is acceptable for us as the assessors and for the EFSA panel. But we also need additional comparators, and single events or lower stacks have been used and should be used, of course, but also negative segregants can prove to be useful. So I think these are the main points with regards to stacks and our comments. Thank you.
Thank you very much, Andreas, and I would like to thank all the three of you, Howard, Annick, and Andreas, for your very clear introductory statements, for your contributions here. Again, we now have the time to discuss this part of the draft guidance on comparators, risk assessment of SECT events. Any comments from the floor with respect to this topic? Start with Boots, please.
I think I will be a little bit in line with Europa Bio, is that we also question the single event to be a prerequisite for the risk analysis, and especially when this single event will never be used for import or cultivation, or will never grow in a field, maybe as a negative, as a segregant. And secondly, concerning the subcombinations and the assessment of the subcombinations of SPECT events, when you're sure that no segregation will occur, and you know there's no interaction between the proteins, is it then really necessary to assess all the subcombinations?
Thank you. Any further questions? Joe?
This is, I think, a response to Boots and also to Dr. Plaisier. We have to consider interactions, and sometimes these may not necessarily be interactions between proteins, but they may have an effect in environmental risk assessment. Let me try to give you an example. Supposing you had a stack with a BT plant and an HT plant. Now, in assessing the effect on non-target Lepidoptera of the BT plants for a single, you may already have found that your determination of the effect may depend on host plant density and its relationship and the balance within the crop and in the margins. Now, if you stack that with an HT and start managing it with the use of herbicide, you may affect the host plant density within the crop. You may also the ratio of the crop host plant density to the marginal host plant density, and that may have an effect on your likely, your expected mortality. So that is an example of well, two things, I think. First of all, the need to consider, at least within environmental risk assessment, stacks and the interaction between events, even though the proteins don't interact. And also it further backs up the need that Antoine Messiaen has already alluded to of taking account of management in environmental risk assessment.
Thank you very much, Joe. Any further comments? Yeah, please go ahead.
Hi, Catherine Goldstein from the High Council for Biotechnology in France, the evaluation agency for France. I'd like to intervene on the issue of tight genetic linkage between three events. One reason is that we have answered in the consultation with a proposition for a better definition, and we had second thoughts about our answer. So I quote the sentence in the document. It says, if events within a stack can be demonstrated to be tightly genetically linked, then by definition segregation of these events can be considered unlikely. And this subject is also in chapter 3.4. It says, should the retransformation result in tight genetic linkage between the new and existing events, then applicants should provide evidence that independent segregation of these is not possible. So first, the question is, what do you consider, or what is the exact definition for tight genetic linkage? And in relation to that is, how does one demonstrate tight genetic linkage? So to answer that, we had proposed first that maybe a genetic distance could be proposed in centimorgan. But we had second thoughts about that, thinking that as soon as you demonstrate a genetic existence that is not zero, then of course you will have demonstrated that they will segregate. So with what we are thinking at HCD is that maybe you should only consider events that would be introduced on the same transformation cassette as genetic meanings and as considered as one single event, or through code transformation where you would prove that two different transformation cassettes would have integrated in the same physical locus.
Thank you very much. A very pertinent question. I think to some extent it already relates to the next topic of our discussion when we will talk about stacked events resulting not from conventional crossing, but maybe we can already start with some answer on this important point now, and then see how we can continue in the next round. So we have three requests from the floor now, maybe a fourth one, and then I give the floor to the podium. Pete.
Thank you, Helmut. To follow up on the points made, whether or not you can use negative segregants as comparator, two arguments have been mentioned. Technical arguments, and with reference to the effects of the transformation, and I can only urge EFSA to keep an eye on the recent research. My colleague Stefan Rausken referred to this morning as to what extent the transformation would actually result in more changes than conventional breeding. That's one point. The second argument that was mentioned just by an Austrian colleague that why you could not use them is because a negative segregant would be a GMO in the definition of the directive. Let me just point out that the jury is still out on that point. There's a working group under the Commission at the moment discussing new breeding techniques, and people look differently at the question to what extent negative segregants and other results of new breeding techniques are actually GMOs under the directive, and that working group is still working hard. The report is being prepared, so it is something to bear in mind. Thank you.
Thank you. So is there anybody on the podium who would like to react to some of the questions raised from the floor? Claudia, please.
Okay, I would take the question regarding the single event being a prerequisite for the risk assessment of stacks. The reason why, well, this morning we have heard, and also this afternoon, that the panel is trying to allow some flexibility in the definition of a suitable comparator in the case of stacks, especially for those cases where a conventional counterpart is not available. The possibility of using lower stacks down to single events, as well explained in the guidance, requires the fact that unintended effects must have been ruled out previously. And the only way to do so is if the single event have already been risk assessed. So that becomes a scientifically sounded prerequisite in order to allow flexibility in the selection of comparator for the stacks. This is a scientific reason. In addition to that, and I'm looking at colleagues from the Commission, in the draft regulation that is not yet finalized as under discussion, along the same, I think, logic, we have a quite explicit requirement saying that single events are a prerequisite for the risk assessment of stacks. And I think, I don't know if colleagues from Commission want to comment, but I think this was inserted by the Commission following exactly the same logic. If we have to allow some flexibility, we have to protect consumer for the possibility of unintended effects, and if we can rule them out before assessing the single events, then we can move forward and open the door to some flexibility to be able to perform the risk assessment of stacks when conventional counterparts are not available. So this is the scientific logic behind that. And then I think, Howard, you can address sub-combinations.
Yeah, with regard to sub-combinations, I mean, the door is also open for applicants to make a written case for why there would be no interactions with the lower level stacks. We're not necessarily asking for experimental data. So we're asking for an interpretation of what might happen if you had lower level stack. So it's not always a data requirement, certainly for the succeed side of things. Can I cover your title, genetic linkage? I think it's a good question. Again, it was to try to give options to applicants in difficult or not so difficult circumstances. In terms of type linkage, in terms of retransformation, and we're kind of jumping the gun a bit because it's the next presentation from me. In terms of retransformation, I think it's exceptionally unlikely you're going to get an event inserting next to the first one. So that is probably not even worth considering. With co-transformation, in some cases you get the plasmids integrating or the cassettes integrating in two sites, but sometimes you can get co-integration. So that is a case you have to look on more carefully. In terms of the multi-gene cassette, okay, you could argue that segregation isn't an issue, but recombination certainly is. So you cannot avoid looking into the option that changes even within that multi-gene cassette could give you some sort of problem, and I'll talk about that later. In terms of how you prove genetic linkage, one of the requirements we have in the risk assessment process is to show genetic stability and inheritance patterns of the events. So if you're carrying out a stability analysis and a segregation analysis and you're finding that the events always co-segregate in that particular study, that's pretty reasonable evidence. It's not complete evidence because you could never prove a negative. But at least it's a priori evidence that the events are linked and do not segregate in the manner in which you would expect them to using traditional Mendelian inheritance patterns. That's one interpretation.
How many meioses?
Yeah, okay, it would be silly to say we want you to repeat this 1 million times, but during the process by which you assess stability and you assess inheritance patterns, any inadvertent pattern that does emerge should be worth additional investigation. That's all I'm saying. Okay? Can I make a question about a response about negative segregates and conventional counterparts, because I think it's pertinent. It's clear, well, hopefully it is clear from the presentations, it is always going to be to the applicant's benefit to find a conventional counterpart. It is always going to be your benefit to find a conventional counterpart. We're just looking at the cases where, according to breeding pedigrees or genetic distance, whatever you want to use, this is not a distinct possibility. So we have to look at options, and I think you've already stated, Claudia, that that's why we look at the singles first, because they've been looked at with a comparative, with a conventional counterpart, but it's also why we have conventional counterparts embedded in the equivalence test. So it's not like we're carrying out any trials without no GM comparators being present.
Okay, thank you very much. Are there any further questions from the audience? Start with Harry, please.
Yeah, to follow up what Harvard said. So there is no difference what Andrea suggested with respect to negative segregants and what Howard just said. Is that right?
Yeah, I mean, what he said is that a conventional counterpart should be used, but in case there is no one, you could use a negative segregant. And this is our problem, because it leaves the possibility to avoid the non-GM comparator. And that's the problem we have. Even if the possibility is a very small one, it is there. And if you have a justification, whatever that is, because that's not specified in the document, that there is no conventional counterpart, you might use a GM. And in our point of view, that is not in line with the legislation. That's the thing I'm saying.
Okay, thank you, Andreas. Stefan.
Yeah, thank you. Just 2 points as a food for thought, maybe for the discussion over assessing the change in management practice associated with the cultivation of GM herbicide-tolerant plants, which I find a very interesting issue. The first point is that for those plants to be cultivated as a farmer in the practice, you don't need to use the complementary herbicide. You can, of course, always cultivate those plants and use conventional herbicide regimes. The second point is we have a plant with the same kind of trait, meaning resistance or tolerance against broadband herbicides also in conventionally breeding platforms. There's the Clearfield breeding platform from the BASF company, and there's also a company in the United States who are just developing a flex variety with a resistance against glyphosate based on a mutation breeding technique, which is very precise, and the regulators in the United States have already declared that those would not fall under the current regulatory system concerning the GMO plants. So we are having the same kind of risks as Joe Perry indicated concerning weed shifts or impacts of the weed shift on non-target organisms, and we are regulating them for GM plants, and we are not regulating them for conventional bred plants, and this is an issue I think should be taken into consideration. Thank you.
Thank you. Any further comments from the floor? Yes, there are two ones. I start with Joe Perry and then Andy Tommy.
I'd just like to ask you, Andreas, how you react to Howard's point that in the food seed compositional trials, because there will be non-GM reference varieties, there is within that trial a comparison being made with the GM and non-GM. Whether or not the null segregant is the whether or not null segregant is used as the other comparator.
Okay. Concrete question to Andreas, so maybe you would like to answer before we move on, please.
I'm not sure if I got it right now. What you're saying is that in the GM food feed, in the molecular characterization, there is always a non-GM comparator, correct? And but as far as I understood it, that's not written as such in the document. If you look at the document, it says always with the food feed and also with the environment, if you can demonstrate there is no GM counterpart, you might use the GM. If it's a negative segregant or a single event, doesn't matter. And I think that's the problem we have. First of all, it is not really any guidance given on how it could be justified that there is no conventional counterpart. We acknowledge that the GM parental line is not, the non-GM parental line is not always available, but there might be varieties which have a very similar breeding history, things like that, which are very close actually to the GM, the stack. And the second one is if it can be justified by the applicant, you allow for a GM comparator, and that's as a sole comparator, and that's the point we made.
Okay, well, thank you.
For food feed and for environment.
Thank you for this clarification. It seems to me that there is maybe not such a big difference in the way how this topic is approached. It may depend on the careful phrasing of the issue, and when revising the draft guidance, this may be an issue the panel and the working group will look at. Yeah, we have two more requests for the floor. I take those two, then maybe short answers, and then we continue. Andy?
Yeah, Andy Tommy, Pioneer. Listening to what Claudia said about single...
Do you have a microphone?
Yeah. Hello?
Yes, great.
Listening to what Claudia said about assessment of singles first, and I think it's already been basically decided that a conventional counterpart has to be used. Moving on to stacks, on the proviso that you've assessed those single events, how does the panel feel in terms of genetic similarity? You can use a conventional variety, but very often in comparison to a negative segregant, it's far, far removed from the GM plant. And so I don't even think it's a balance. Obviously the prime goal is to prove safety, but we can't get away from the fact that it is there is always a comparison. And I think this, the genetic similarity itself has to be considered.
Okay. I take the other question for the floor of the gentleman to your right, and then we have an answer from the podium.
Pioneer DuPont.
again, on the use of negative segregants in the stack comparison. So this is the nature of the seed business. It was mentioned today several times, licensing in and licensing out of different events. When we buy an event, in a stacked event, in our stacked product, we've got the seeds and the discussion of the generation as BMSN, where the M should denote the number of backcrossing and the N the number of self-crossing. So actually we don't know anything about the breeding history of the event we buy. So even we would like to be in line with the requirement to provide the most appropriate, most closest known GM comparator. We just simply don't know. Imagine if we have a four-way stack, a quadruple stack. We buy 2 of the 4 events from a comparator, for a company. We don't know anything about that, about the breeding pedigree of those events. How could we establish the right no-GM comparator for such situation? And my question, anyway, that whether such a situation would justify a convincible justification for the non-existence of the comparator.
Okay, thanks for your questions. Is there any reaction from the podium on that, or shall we take it as it is, as a comment, maybe discuss it also in the context of our next round of discussion when we continue with the fact topic, but move on a little bit further. So, Claudia, would you like to react now? Or?
No, well, very briefly, I'm repeating a bit what was said before in reply to what Andy said. I think that what we need to, I think sometimes we tend to confuse things. We are talking about non-GM reference varieties in the equivalence testing component of the comparative assessment. And the difference that the panel is allowing in terms of selection of comparator relates exclusively to the difference test component of the comparative assessment. Non-GM reference lines will always be included for all of them, single and stacks, with no difference. In that respect, when we focusing only on the difference testing, the hypothesis of opening the door to negative segregants, which has to fulfill certain criteria which are listed in the comparative document, allow for a comparison of difference with a line that is genetically close to the GM. This, however, has nothing to do with the non-GM reference varieties that must include anyhow in the field. So I think we should clarify that confusion, as they will always be included. And this has been addressed in different ways also by Andreas before. So it's a recurrent confusion that we are discussing here.
Thank you very much, Claudia. I think this is an important piece of clarification. Sebastian, briefly.
Maybe a short reaction on herbicide tolerant non-GM crops, because it was stated that it would not be assessed at all. It's true that if it's non-GM, it doesn't fall under the GM legislation, but it would still fall under the pesticide legislation, so all the environmental safety assessment and the use of the herbicide would fall under the pesticide legislation and would need to be assessed before we authorize its use.
Okay, thank you very much. Very briefly, but then I want to move on, please.
Yeah, just as a reaction to that, but that does only cover the immediate action of the herbicidal compound that is used and not on the change in management practice, which relates to timing, when the farmer uses the herbicide and the weed shift that occurs, and that is not covered by the current pesticide regulation. So the wider ecological impact of the change in management practice is not covered by that.
Thank you very much. I think we should not move too far now into the pesticide regulation area, because we are focusing on a very concrete topic, which is very good that we do so, and this is complex enough. Thank you very much. I would like to thank all the 3 speakers of this set, and we again have a slight change of the podium. Howard can stay, but Inge and Jan are joining the podium. Okay, dear colleagues, we are now moving on to the third set of our discussion this afternoon. We are still with the topic of stacked events, but this set is dedicated to stacked events not from conventional crossing. So we are again going to hear a brief summary of the comments received on that part of the draft guidance by Howard, and then we will have two invited comments by Professor Inge Breuer from the University of Rostock in Germany, and by Jan Pedersen from the National Food Institute in Denmark. So I would kindly invite Howard to start. Please.
Thank you. I think this is a wonderful opportunity just to carry on the same discussion, because it's the same points in the singles, the stacks, and this type of approach for stacking that will come through, I'm sure, in terms of the debate. This slide basically shows us the various ways we can stack genes. We can conventionally cross one event with another and end up with a two-event stack. Or we can retransform an existing line to add a new event, or we can use co-transformation to increase the number of genes that we're inserting at any one time. And I think I've already covered your point about linkage, hopefully, and that won't come up again. I think there will, at some point in time, be more of a move, I suspect, towards stacking genes, and I use the term stacking loosely, within a multi-gene cassette. There was this paper that came out recently by Azadi, who actually put 7 genes encoding 7 different enzymes together in the carotenoid pathway. They all expressed and they all had an impact. So this is another way of stacking genes without the concern of actually crossing like with like and worrying about singles. Although, as I mentioned just now, the issue of recombination should still be assessed when looking at the potential impact of loss of function of any one of those genes. So, linked or unlinked, how to prove, that was one of the comments that came up, and we discussed that in the last session. Again, the issues surrounding the use of a GM as a primary comparator. And also many of you commented that all GMs, however produced, are best assessed with a non-GM comparator. That's coming through certainly in the debate we've had this afternoon. And in terms of multi-gene cassettes, as I've already said, you still need to consider the potential for loss of function within those particular GM events. That's all I really wanted to say, because I think a lot of the issues are similar that we're covering just now.
Thank you very much, Howard. Very clear introduction, and I would now like to invite Inge to give her invited comment, please.
Yeah, thank you very much for the invitation. I'm really surprised, actually, to be invited, because I'm not from the ministry, I'm not from an organization, I'm just a university professor working on transgenic plants and risk assessment since more than 20 years. And as such, I'm a coordinator of a huge joint project where we try to develop methods for sound risk assessment and effective risk assessment. So as such, since more than 20 years, I'm looking for the right comparator for my experiment. And what you do normally is that you say, I have a transgenic plant, or I have a plant that I would like to look at, I have a question that I would like to ask, and then the question is normally, what does a certain protein do in a plant? And then the comparator is normally the plant that is absolutely similar, with the exception that the protein is not there. So this is the scientific sound thing to do. It's just one factor change. So do it in that way, and you have stacked events, then the plant that has not the new gene, or if it's a transgenic original plant, is the right comparator. No question at all. For sure you have the conventional counterpart that is very similar to the transgenic original event, but it is different in 2 factors, so it's not the right comparator in the primary choice. But as we all know, it is important to compare to other plants, so it's also important to compare to the conventional counterpart, and you would accept more differences in this comparison. And then you can judge whether the differences between the transgenic event and the transgenic stacked event is due to the new quality, or whether it's due to the conventional counterpart. So you have a lot of questions you can ask then that you can't answer when you use the conventional counterpart as the primary comparison. I would really like to say that it's important to use other conventional lines in addition. So, since the approved events are considered as safe, they should be the counterpart in the comparison. I really wonder why the GMO panel hesitates to do that. Because if they say these plants are safe, then they should use them. If they don't use them, they say they are not as safe as the conventional counterpart. If this is true, then you are not able to use them or to release them. So if you want people to believe in what you're saying, then you have to say, okay, if we assess it and if it's then really safe, then we can treat it like a conventional plant. So this is what really amazes me in a way. There was a second point that I really don't understand completely, where I think we should have a look at, is when you say that, for instance, for higher amylopectin transgenic event, it should be good to use the higher amylopectin conventional variety as counterpart. This might be interesting if you want to know what the transformation itself does. But normally, in the evaluation of transgenic plants, we want to know what the new protein does in the plant. And this is the main case, because I think we all know that transformation itself is not a risk. And this is what comes up here all the time when you talk about non-transgenic segregants. What should be the problem with these plants? You have assessed the transgenic line already, so you know whether there are other copies of the gene or other gene fragments inserted. If they are not present, then this plant is genetically identical to the conventional counterpart, except all those rearrangements that might have happened during the regeneration protocol, so just the tissue culture period. And this is why the non-transgenic segregate is the real control, because it's more similar to the transgenic plants than the non-conventional variety. So this is why I think, from my just simple, non-political, scientific point of view, you should really try to find a comparator that is as near to the transgenic plant as possible, so that you change only one factor. That's all I wanted to say.
Thank you very much, Inge. And we move on, and I would like to give the floor to Jan for his comments, please.
Yeah, and I would like to thank for the invitation to give some idea of the Danish comments. And I have to apologize for this presentation might not be fully representing the comments from the environmental side of the Danish comments, but I think most of them will represent them also. Yes, I need to work by this. We are speaking about 3 different methods for creating stacked events. And the comment from the Danish side was that we don't think there's a need for making distinctions between these 3 different methods, but instead saying what is the result of these methods. And as you see, there can be a linkage of the genes, and that should be taken into consideration. One of the traits could be already assessed, or maybe it's not assessed, none of them. And varieties for the host and the back crossing can be different from these events. And I also want to point out that when we are speaking about traits here and genes, we are discussing a wide range of different constituents, genes, and traits. It could also be mutation made by GMO techniques, in fact. So that's why we can see the retransformation might be a second transformation of a GMO where we could put in a gene close or make changes in already inserted genes from another GMOs. And our proposal is that if there is a linkage between these traits, we should use the closest non-GMO for comparator. That could be one of the solutions. One special case perhaps is the retransformation, because there we already know there has been a first GMO that has been transformed again. And I've divided up in two situations. One where the first one is already approved or assessed, perhaps we could stick to that, or if neither of them has been assessed. And I mean, maybe it's what's going on in the lab. We have the first GMO, and we go further on, or in the greenhouse, go further on with the transformation with the trait B, and then we went up with a GMO A plus B where none of them has been assessed before. And I think there we can use it could be a GMO comparator A, or if we have this available, or non-GMO based on the closest genetic background. And of course, in any case, the assessment always involves the use of data from the approval of the single GMO A, if that is available. In about this, where we have this the linkage, as I mentioned, there could be a linkage between the traits. The comparator could be for, as we have for single events. If there is no linkage, the comparator could be, as we know, for single events, and either the GMO A or B. And I like this flexibility that is put into these comparator guidelines that we have, and I want to and since I like it, I want to say that could be the way to do it, but there might be exemption from this, case by case, meaning that in some cases we don't need to evaluate the GM A or B, or maybe we need them both. So based on scientific experience that we have today, there could be situations where we don't expect interactions, and that has to be taken into considerations. For instance, the A and B traits could already be put in another plant, in maize, in soybean, and we have experience from that. We should use that knowledge also. So, in fact, the conclusion is that we can use this same approach for all these three different methods, and take care of whether they are linkage or not, and we have had a discussion about linkage. And my final question is here, is there any scientific justification for handling the transformation of an approved GMO different from the transformation of a non-GMO? And we have heard that earlier today also. Thank you.
Thank you very much, Jan, for your input, and I would like again to thank all three speakers for their very concrete presentations. Again, now this is the possibility for you to raise questions or make comments. to this part of the guidance. Stacked events not resulting from conventional crossings. Susanna.
Thank you. I would like to comment on Inge saying that transformation itself is not a risk. Well, as far as I know from the literature, when a gene insertion occurs, then in the genome, 2 to 5% of the genes, gene expression and function is changing. And that's what's causing unintentional effects. So I can't agree with you. I'm sorry.
Thank you. Any further comments? I again collect a few comments from the audience and then give it back to the podium. Any further comments, questions, reactions from the podium? Yeah, please.
You know that quite a lot of things, rearrangement happen in the genome of a plant normally. Transposons are jumping and a lot of other rearrangements occur in the genome normally. So the genome is changing all the time in its structure itself and in its expression. So when we try to compare the expression of genes in a top leaf of a plant and a leaf further down, we see enormously different things. So it's absolutely impossible, really impossible, to make experiments that can show you that due to the insertion of a gene, 3 to 6% of the expression is changed, because the expression is changing all the time dramatically. And to do an experiment where you really could prove that statistically significantly, I think it's really not possible. So it might be that someone is publishing that. But when you really look at the variety and the variability of gene expression in plants, then you will truly see that this is something you can't say. And when you look at, I analyze enormously many transgenic plants and non-transgenic plants due to their variability. And you never find significant differences between transgenic plants and their comparators when you look or compare these differences to the differences in other transgenic or non-transgenic plants. In all cases. So for sure, the integration of the transgene itself doesn't do as it is a harm. For sure, if it integrates into a gene or it is regulating other neighboring genes, no question, and we have to look at that. But the process itself is a totally normal process in plants. Jumping genes and jumping fragments of chromosomes are normal.
Thank you, Ian. Of course, we are entering now in the area of our discussion which is more of a general nature, transformation, risks resulting from genetic modification or not. This is always interesting, I understand, certainly, but probably what I would like to recommend is that we focus on the topic of our discussion this afternoon, and that is the selection of appropriate comparators for this specific topic we are discussing right now, and that is stacked events resulting not from conventional crossing. But I saw 2 requests for the floor. Stefan first, and then, okay, 2 or 3.
Yeah, if it's okay, I wanted to first weigh in on what Inge just said in response to that question. There was this very nice publication by Batista et al. Who showed that genetic modification actually caused fewer changes than different mutagenesis techniques that we are currently using in conventional agriculture and have been using as a conventional technique in plant breeding for the last 6 decades or so. So if we consider changes in the genome or in the transcriptome as relevant, we would need to assess that also for conventionally bred plants. And again, here's the argument that what is statistically significant in terms of up-regulation or down-regulation is itself not something of concern. The question is whether that is actually biologically relevant. And Inge already said we have a very large biological variation depending on whether the sun shines or it rains or it does not, whether there's enough phosphorus in the soil, manganese or whatever, and these changes are quite profound, and that's what the question is. The other thing is, I think this is this is not only a scientifically interesting point, it's also a very relevant point when we think about the assessment or the selection of comparators, because it's the strong argument that Inge put forward that we need to find a plant that is, if we want to have an immediate comparator, we need something that is closest to the genetically modified plant, whatever that is. and if that is in fact another GM plant, we should use that as a comparator as well. Thank you.
Thank you. There were two other requests for the floor. Klaus Ammann first.
I just want to support Inge and Stefan Rauschen. And although it's not the topic, but actually it's related to the topic, that natural mutation and transgenesis have the same three molecular processes, and that has been published by Werner Arber in the 50s already. So it is common knowledge that the transgenesis and natural mutation is on the molecular processes level exactly the same. It's not different, and that's one of the big mistakes in regulation, which was not done in the Canada system. I just want to repeat this. It's not only Batista, it's 40 genomic analysis papers which have been summed up by Ricroche in a very nice recent paper, and it's clear that we have to take that into account in future regulation processes discussions.
Okay, thank you. Right now we are discussing the selection of appropriate comparators, and there was a request for the floor, please.
Sławomir Sowa, Plant Breeding and Acclimatization Institute at National Research Institute Radzików in Poland. I'd like to stack all the stack sessions in one. And this is kind of an open question. What should be used as a comparator for the risk assessment when the genetically modified stack variety results from a very complex and sophisticated breeding program? Namely, if we have a crossing of a single GM genotype, retransformation, co-transformation, and transformation with multi-cassette genes at once, in one variety. I think that breeding used to be about crossing, back-crossing, inbreeding in the past, for many generations also, before commercialization. And now breeding becomes a much more complex process. So new breeding techniques, we have oligonucleotide, direct mutagenesis, reverse breeding that can be combined with the transformation also. So the more GM varieties will be introduced into a breeding program. We had information from the pioneer company that sometimes we can get, let's say, breeding material from another breeding company with a very limited knowledge about the source. So those varieties will be introduced in the breeding schemes. And I believe that we might face such a situation. Thank you.
Thank you very much. You are not making our lives easy just before the coffee break, but would be interested to hear some reactions from the podium. Please.
Well, you could use the parent to look at the parent, to look at the parent, to the parent. It's a cascade effect of safety. You could think about it that way. Or you could take this wonderful plant that you just described with all of these events, cross it to a non-GM plant and use the negative packaging.
Okay. Any other requests for the floor? Yes, please.
I am Barnabas Janes from Hungary, from Agricultural Biotechnology Center. I'm just having a short comment on Jan's contribution. When we do co-transformation, we usually do not expect linkage. The reason, for example, why we use co-transformation is that this is the way, the segregation is the way how we can get rid of the unwanted selection gene. Okay?
Thank you very much. Further questions, further comments? Need for reaction from the side of the podium?
No, I would agree in principle that the literature does say that you can actually get co-integration of these things happening. So if you're looking for stacking genes and you don't really care whether they're one event or two events, you could actually potentially use co-transformation to insert genes at a single locus. So it does happen.
Okay, thank you very much. Fine. If there are no more requests for the floor, I would like to thank again Howard, Inge, and Jan for your input. Thanks to all of you for your reflections and discussion points. I think we all deserve a coffee break now, and I would like to hand over to Rita, please.
Thank you, Helmut, and thank you for the speakers and audience. So we had a short coffee break. I would like to ask you to be back at 4 o'clock since we have sugar at the bottom and there's no comparator. What shall we do? So please come back at 4 o'clock. Okay, so welcome back to our last part of this workshop. And without due course, I give floor to Helmut so we can have the final session starting. Please, the floor is yours.
Thank you very much, Rita, and also from my side, good afternoon to all of you. Hope you enjoyed the coffee break. And as Rita has said, we are now approaching the last part of our workshop today. We will deal in the next 35 minutes with the fourth and final chapter of the draft guidance on comparators, which is about risk assessment when no comparators is available. And here we will have a summary of the comments received during the public consultation, which will be given by Dr. Harry Kuiper, the chair of the EFSA GMO panel. And then we will have 2 invited comments on that topic. First one by Dr. Christoph Kane, Executive Director of Test Biotech. And then another invited comment by Dr. Boos Klandorf from the National Institute of Public Health and the Environment in the Netherlands. And then we will have a discussion. So I would like to ask Harry to give his presentation. Please, 5 minutes.
Okay, thank you. In order to get rid of all the discussions on substantial equivalence, and Peter Kunz might like that, I would like to introduce the approach on risk assessment without any comparator, which is actually chapter 4 of the document. I'm not very happy with the title of the chapter 4 as it is now, because it says alternative approaches, as if you could use that approach instead of the classical comparative approach. So we have to think of a better title later on. The issue is, there may be cases, and I've just mentioned here a few, and Howard did it this morning, where we are faced with products which are extensively changed. And can you still use the non-modified one as your, I would say, classical or conventional counterpart? Now, in some cases you may be able, and in other cases not. So I think it depends on the extent and the complexity of the genetic modification. That determines, or that indicates whether a comparative assessment in the classical sense is possible or not. If it's not possible, then the guidance document says you should do a safety assessment per se, whatever it means. Some people say you should do a full safety assessment, as if a comparative assessment is not full or half full. So we have to be careful in the wording. But one thing is clear, and that is probably something for the near future. We need further detailing of the safety assessment strategy per se. There were 3 main points, I think, from people, member states, or other organizations who reacted on the documents, which I think are worthwhile mentioning, and we have taken them into account. The first one, which is here, comes from, I think it was Austria. And I think that is a fantastic good remark of Austria, by saying it is not so much the fact that a conventional counterpart may not be available, but it is more what is the strategy and what are respective studies to be done, to be provided in order to prove the safety. I think that is a very right observation. Furthermore, in the document, we had an example of a GM, in this case oilseed rape, where We stress the fact that you may want to look at other comparators in addition to the traditional one. In this case, it's what about lauric acid, and you may therefore look at other products which contain lauric acid, palm kernel oil or coconut oil. But the mistake in the text was that we suggested this as the only comparators, which is not the case. You, of course, should look at the principal comparator, which is the non-GM oilseed rape. And then there was a paragraph on unintended effects of covered in the environmental risk assessment, and we have taken that out because I think that is covered in the ERA guidance document. Now, and to close with, when it comes to a safety assessment per se, whatever you want to call it, there is a list of issues you should look at, and I've just listed here a few. And I think the main body of that assessment is, first of all, the type of modification. Secondly, and that will become even more important, is an extensive compositional analysis with preferably the best modern and newest techniques available. With respect to toxicity, you should look very carefully at all the components present in that food, and we should refer to threshold levels, ADIs, etc., to get a better idea on the toxicity per se. And the same applies to allergenicity. And dietary intake, of course, is also important. I think we have the best available from a toxicological point of view. And to conclude, I do not think there is a conceptual problem with looking at GM crops from a food feed safety point of view when there is no competitor available. Chemicals in general are judged upon by the characteristics as such without using any competitor. We have the analytical tools available. And last but not least, I'd like to emphasize again that a product-based assessment is preferable from a scientific point of view rather than a technology-based assessment. Thanks.
Thank you very much, Harry, for your very clear introduction to the topic. I would now like to invite Christoph to give his invited comments. Christoph, you have the floor.
Thank you very much for inviting me. I'm from a small NGO which is dealing with which is independent from interests of industry in dealing with risk assessment. We did several reports you can find in the internet, no problem, everything is there. When I come to the topic which is here on the agenda, no comparator available, I want to take one step back and ask what's the general problem with the details we are discussing in here. And when I'm listening to many contributions from the audience, I have this, I'm not feeling really happy with the discussion so far, as it was conducted on the comparative risk assessment. Well, in general, I would say we have a certain risk that we are going to the same trap as the tobacco industry was leading the scientists, the scientific community in their sector. And there are three things which are very relevant in this context. Denial of specific risk. What we've heard extensively, there's no specific risk in GMOs. Secondly, influencing scientific standards for risk assessment. I think comparative risk assessment is the closest risk assessment, or let's say the risk assessment that is closest to the interest of industry. And so it's close collaboration with scientists and international institutions. And the general problem we are facing with the comparative, that we might choose a wrong comparator. This is the general problem in here, and this might lead to effectively a masking the specific risk, and not talking about specific risks, but just about data which are not relevant. And this is, for example, relevant for the risk assessment of golden rice. Of course, this is taken from the homepage of the golden rice. And the other example was already mentioned. It is there in the draft guidance, and we already heard it was a wrong explanation, was given, or wrong wording. It's not meant in this case that rapeseed should be compared with coconut oil, but the general problem is there, how to do a proper comparison, and not only what is the right comparator, but also how to use the tool of comparison in the right way. These are two different questions. One is what is the comparator, and what can really the tool of comparison, what can it really deliver? And we already heard from the audience that there is no specific risk, but I ask you to take into account that genetic engineering, of course, is dealing with specific technical methods which are not there in conventional breeding, not in mutation breeding. They are different. These have to be accepted. They are patented. There's a new technology, and this technology has a specific potential for escaping or disturbing the process of normal gene regulation. And this specific potential is unique for this particular technology, and we should not deny this specific risk, or let's say the specific quality of the technology. And so in general, we think we should not try to mask the risk, we should try to avoid masking the risk, hiding the risk, and really ask what are the specific risks and develop appropriate tools to deal with this risk. And then now it comes to the question which is now on the table: what can we do if we do not accept that crops derived from genetically engineering are compared with conventional breeding, or as you say, if no comparator is available, what can we do then? And we have a very clear idea what a risk assessment per se could be. It's a very easy, let's say, introduction to another kind of risk assessment, which I don't think is really technically a big issue. I think it can be easily implemented, and should meet all the needs from the risk assessor and the risk manager at least. So what is already done is the molecular characterization. This is already done by EFSA, but what is missing is something like a stress test, really to see how the genetic stability is interacting, or if it's there under stress conditions, how does it react with the other genome. This is missing, and this could be a very important element of a risk assessment per se, assessing the dynamics of the modified, of the genetically engineered plants. And the second thing of the stress test would be to try to determine the range of variation in the compositional analysis, not only having it at one time, trying to make a comparison between the conventional plant and the genetically engineered plant, but really to look what is the plant doing if there are certain conditions like heat, like drought, how do the composition of the plant, how do they change, what is the range of variations which can be observed in the plant. This is highly relevant in the case the plant is producing insecticide, for example. You want to know what are the variations. This is not known so far, and it's not required. And then in toxicity, we think we should start in the lab with in vitro investigations, which would mean we can have new proteins being tested, or whole plant preparations being tested, for example, on cell cultures. This is done to some extent already, but what is really missing is a systematic approach to test the combinatorial effects in the laboratory before we go out in the field, or before we do a second step in risk assessment. So, for example, of course, I want to know what about the residues of the pesticides being sprayed on the plant coming together with the insecticides produced by the plant. This is done to some extent by industry, I know it, on target organisms, but as far as I can see, it's not done on non-target organisms, or not related to human health. So this is really missing, and this could be overall a starting point for something like a risk assessment per se. So when you compare it to the risk assessment which is done so far and come to a risk assessment per se, it would mean you have another starting point. You do not start with a comparative risk assessment. You start with generating a broad range of unbiased data, and then you make your first hypothesis, and then you develop maybe case by case what the other steps should be during a risk assessment. So it's just having it as a starting point. This is the central issue, the central message. And the second thing is you should have then a proper hypothesis, and then you should have more mandatory testings, experimental testings, like multi-generational feeding studies. In case you want to go forward with the risk assessment, you should ask for multi-general feeding studies. and you should ask for experimental releases in all biogeographical zones over several years, going step by step from small scale to larger scale. So last slide. We would propose to drop the concept of comparative risk assessment completely. We should not presume safety, equivalence, similarity, or familiarity. We should use the comparison just as a tool and not as a concept. We should always apply risk assessment per se when it comes to risk assessment of genetically engineered organisms. So thank you very much for this, for your attention. Clearly, I was a little bit out of scope, but some of the issues I was putting into the discussion, I think you can also use for your specific question, which was given to me when I came to this meeting. Thank you very much.
Thank you very much, and I would now like to invite Ruth for her invited comments. Please.
Good afternoon. First of all, I want to congratulate EFSA with their guidance. We think it's a good initiative. We have, however, of course, some comments which I want to elaborate on. First of all, I'm from the GMO office, where I'm responsible for handling all applications for GM plants, also for the environmental risk assessment, and we do that for the Ministry of the Environment of the Netherlands. And in the process, we also ask advice from our Committee of Genetic Modification, and today I represent both the Ministry and the Commission of Genetic Modification. And we filed several comments, but I only want to stick to the scope of this session. And this is about when no comparator is available for certain GMs or when other approaches should be necessary. And I have two comments actually, and we touched already a couple of times on them today. And it's mainly about this section in the guidance of comparators. And so it's mainly about the environmental impacts. And I highlighted the sentence that the comparator should be a non-GM line that is as close as possible to the characteristics of the GM plant, except for the intended modification. And of course, you can imagine that for certain GMs there might be a problem there. And the first example is, which we already discussed a couple of times before, you can have modifications which are quite complex, which affect the metabolic pathways in plants, and there can also be crosstalk between pathways in plants. So that leads to complex modifications. And the question here is, what to use as a comparator for ERA in this case? What is the best comparator to use? So that would be my first question. And the second question is, we also touched upon this, but that has to do with new traits like abiotic tolerances. There you can have GM plants which have a non-GM comparator. However, the GM plants will be grown in new areas, like the areas where there's a higher level of salt or more drought. And the non-GM comparator is unable or maybe unable to grow under these conditions. And since the environmental risk assessment is based not only on the comparator but also on the receiving environment where the GM is grown, we really have an issue here. And also the question here is what to use as a comparator in this kind of cases. Is there actually one comparator that you can use, or do we need in this case another approach? So I want to leave it with that. Thank you.
Okay, thank you very much, Ruth, for your invited comment. I would like to thank all the three speakers for your very clear presentations. And of course, Ruth already raised two questions, which probably require some kind of answers, but still I would like first to give the possibility for additional questions from the audience? Stefan, please.
Yeah, thank you. I just wanted to comment on a few things that Christoph Tim mentioned in his talk, because I think scientifically he is so far off base, he's not even in the ballpark anymore. Because a lot of these issues he's raising are already covered in what the EFSA is doing in their risk assessment and what is also done by public researchers all around the world. In my own research program, we have been assessing the expression of Bt proteins in now 3 different Bt maize varieties over 3 seasons each in different locations. So we know very much what the variability of expression of Bt proteins in Bt maize actually is. And the question, if this is relevant or not for the environmental risk assessment, is what are the limits of expression and what is the actual exposure of non-target organisms in the field, and whether these non-target organisms are actually susceptible to the toxicity of these Bt proteins. And as long as that is not the case, it's not really interesting to know what the variation in expression of these proteins is. The thing with getting a lot of data on the plants, on variability of these plants in gene expression, it's interesting, but it just turns the whole system of doing scientific research on its head. First, you create a scientific hypothesis that you know you can test with an experiment. Then you generate data to test your hypothesis, and then you either reject your hypothesis, or if you can't reject your hypothesis, you look at another way of trying to test it. That's the way you do it. You don't just gather a lot of data and then try to sift through the bunch of data and try to identify risks that might be potentially there. And this leads me basically to the question, what the specific risk of genetic modification in plants is that we are hiding with a comparative approach. I don't get that. Thank you.
Thank you very much. Again, I would kindly ask that I know that some of the presentations have also been a bit beyond the scope of this particular set of discussion. Risk assessment if no comparators are available, but I would like that we focus our discussion still on that topic. Inge and then Klaus.
I try to stick to it, although it's really difficult. But the first question I would like to ask to Christoph Thien is what might be the effect of this combinatorial effect, and isn't it necessary to first find out whether there might be a combinatorial effect before you test quite a lot of different plants? And then I have to say something really as someone doing field trials. I'm always having fences and a lot of people to protect the field trials. If you really want to have such a lot of field trials happening, as you said, then please go together with all your NGOs and help us to protect these field trials from those people that in your name try to destroy them.
Thank you.
Not in your personal name, but in the name of the NGOs.
Can we still try to stick to the subject of our discussion? I would really appreciate if we are able to do that towards the end of the day, because I don't think there's much we can gain when we discuss on such a general level, and I would also be very happy if we can discuss with respect to the tone in a way so that we respect each other as colleagues here in this room. Klaus?
Yes, I understand your concern, Helmut, that we should stick to the topic of the stacked genes, but the things we say here are intricately related to what follows up for the regulation of stacked genes. If we talk about the genomic misconception, natural mutation the same as transgenesis, it is having a follow-up on the stacked gene debate quite clearly. So you cannot just separate it and say, well, we discussed that next year or something like that. Because there's also another misunderstanding. When I now stress this topic, I don't mean that we don't need regulation, but we need a differently organized regulation which is based on novelty and a corrected approach like the Canadian system. And that has an influence on everything in this room is debated. And you cannot just say this is not relevant to the whole debate.
Thank you very much. Beatrix.
Thank you. So I'd like to come back to the assessment per se, on the problems in the assessment per se. And I think it would be very good if there is a little bit more of guidance in the document or in the opinion of EFSA. And maybe it is possible to learn from other fields. For example, there is quite a lot of experience in the context of ecotoxicological testing. It has been mentioned by Harry Cooper that there are tools available. So they have to be adapted to a certain extent, because when you test chemicals, it will be a little bit different when, from testing of whole organisms. But maybe we can look into these areas in order to learn how to do such an assessment. And there may be also the possibility to adapt it to a certain extent what has been laid down in the environmental risk assessment opinion in the context how to choose the right at that place non-target organisms of different functional groups and all these aspects. When you come into environments, for example, or you have GM plants where there are no comparators available, you may think which are the relevant organisms. For example, when it's an environmental assessment, which are the relevant organisms in the receiving environment, which are the functional group, what are the ecosystem services and the relevant organisms into these contexts. And then you try to develop or adapt what is in ecotoxicological testing in order to find out what is the impact on these functional services, and that maybe inform what is then the decision in your assessment.
Okay, thank you very much. Are there any reactions at this stage from the podium? Christoph? Briefly, please.
Okay. So, thank you for your questions. I'm talking too fast, so maybe you did not get all details I tried to introduce. Sorry for that. On the BT concentration, it's very fine you're doing these testings, but what I said, we need a requirement from the risk assessor that this kind of testings are done and the data are published. And this is so far it's not really done. Not even a method is available that would allow to compare the BT measurements of measurements from one lab to the other, because the protocol is not defined. So there is some things which really have to be done, that we can have properly assessment of the variations of the newly produced proteins in the crop plants. And when it comes to how to develop a hypothesis, I agree with you that as soon as you want to have a certain risk to be developed, then you have first to develop some kind of hypothesis and go there and try to test it. But what I was talking about as a starting point in risk assessment is to try to get more data on the technical quality of the plant. How does it react to confined conditions, to stress conditions? How is the genetic stability being performed when it comes to certain conditions, interactions with the environment? And maybe the thing which was or the last question I can answer, because the rest would take too much time maybe. When you were asking what kind of risks are hidden, I try to expose the example of the golden rice, where the risk of eating genetically engineered rice is being compared with the risk of eating a carrot. And of course you can do this kind of comparison at a certain level. When you want to compare the amount of beta-carotene, then you might apply this kind of comparison, but of course this kind of comparison cannot be a risk assessment. I think we all agree on that, and if you choose the wrong approach in the comparative risk assessment, you will end up with this kind of problem.
Okay, Harry, and then Joe, and then I think I'm going to conclude this session. Harry, please.
Well, on this last point, in terms of too high risks, I mean, the GM rice is an example where initially the bioavailability was not very high, and that was recognized. And a lot of work has been done in that area to raise the bioavailability. So you cannot say there was only a comparison with a carrot to solve the problem. So that's not hiding a risk, it's identification of a risk which then is further assessed. And if I may, a question on what Christopher then calls stress tools. I understand in terms of expression of new proteins, but I thought the subject was here the situation of no comparators. Now, how would you relate the use of stress tools, which I think is doubtful, it depends on the hypothesis you have, in terms of risk assessment when there is no comparator? That was the question here.
Brief answer by Christoph, and then Joe, and then we conclude. Please.
Well, if you have no comparator, you have to try to come up with a risk assessment per se. And my idea is then you should start, or our idea is that you should start with finding out more about the specific technical quality of the plant. And a stress test can help you no matter whether it's a stacked event or a single event, it doesn't matter. You always can start with comparison of the plant's components and gene activity under defined conditions. I would prefer to do it in the lab, so you can have specific questions being tested. And the analogy would be to all kind of technical fields, like a new car, you want to know how the brakes, to which speed you can drive a car without being in danger, and you will test the brakes. And this kind of general investigations to find out more about the technical qualities of the plant would be very helpful if you're trying to find out what a risk assessment per se could be, because this is not depending on a certain kind of comparator.
Okay, thank you. Joe?
3 brief points in response to what's been said. Boots raised a very good point, very good question. She says, is there just one comparator? No, I don't think there is. I think the comparator for ERA should be on a case-by-case basis. Sometimes it might be the closest one can get genetically. Sometimes it might be the closest one can get in terms of characteristics or traits. And I think I would accept the criticism that maybe we had been unclear that flexibility was necessary and that we might need to look again at the wording. Certainly, for ERA, it's always on a case-by-case basis, and we have many different, as I've said before, many different questions to answer. Each question may require a different comparator, so I would not like to suggest that there would only be one comparator. Second brief comment regarding BT and the differences in expression between laboratories. Yes, this is an important issue, and it's something that we need to take account of in our ERA. I believe we do take account of it. I believe we think of worst-case scenarios, and in doing that, we allow for this additional variability. It's not the only source of additional variability, but it is one. That point is accepted. With regard to the being close to the interests of industry and presuming equivalence, I think it's worth pointing out that we don't presume equivalence, we test equivalence. But there's something about the equivalence test that I hope, Christoph, you would be quite pleased with. And that is that in normal biological null hypotheses, we set a significance level, and that relates to the producer risk, the risk of false negatives. That's the normal way one does it. And the consumer's risk, in that case, is difficult to set because it relates to the power, which is difficult to set ab initio. However, in the equivalence test, it's usual for a null hypothesis to be framed differently. There, it is the producer that the consumer's risk is set, and it's the producer's risk which is related to the power, which actually induces applicants to do experiments with high power. It's in their best interest. I think this is a good example of the way that both in environmental risk assessment and in food feed risk assessment, the EFSA guidance documents are moving forward to properly reflect consumer risk.
Thank you very much, Joe. With this, I would like to conclude our discussion on this fourth chapter of the draft guidance on the selection of comparators. I would like to thank our three speakers for their presentations. Thanks to you for your contributions. And with this, we conclude the whole discussion for today on the draft EFSA guidance on the selection of comparators. And in fact, we are moving towards an end of today's workshop. I would like, at this stage, and according to the program, just provide a short summary from my side of the topics which we have been discussing today and the way how I sense or assess the discussion. This will be certainly my personal summary at this stage, and I will try to be brief. And I will also try to take into account topics which we discussed just right now. This is always a bit of a challenge, but I will try to do my best. Well, first of all, I think it proved very useful to have this, so to say, special opportunity to, in addition to a public consultation, to also have a workshop on a very specific topic, on a specific draft guidance, the draft EFSA guidance on the selection of comparisons. I think throughout the day, in my point of view, we had a very open and constructive debate, which was focusing really on the topics of this workshop. And we had, Rita and I said it in the beginning of this workshop, two different but related topics for this workshop. More a general framing of the risk assessment, and then in particular the draft guidance on the selection of comparators. The issues are interlinked, and in fact, from my side, I must say, I think we had a very good, a very rich factual and scientific debate on those issues. This is really a value, and I think this means a lot of progress in this area. From my point of view, it also turned out from the comments you made and from the discussion which we had that the context of the risk assessment framework is very important. We were discussing a very specific document here on a very important topic within the area of risk assessment of GMOs, but the context of the whole risk assessment framework is very important. And I think it also, at least it became clear to me that this context and the whole field of GMO risk assessment is evolving by experience of all of us. It's not a static issue, and it's not only the case that there has been evolution since 1993 and some crucial OECD documents and the year now, but it's constantly evolving, also with right now and within the foreseeable future. In my point of view, as I sense the discussion which we had, I think we identified that concepts, principles, terms, yes, they are important and they have their relevance, but it seems to me that we as a group here seem to be even more interested in what these concepts and principles and terms mean in practice, meaning when we implement them in practice, when we do and assess a GMO risk assessment. Within this comparative approach, a term, a concept which appears quite often during this day, I sense that our common goal is how we can ensure a scientifically valid and relevant design of the tests, of our tests, of the tests to be done, which allows to use the most appropriate comparator as a basis for the assessment. So again, this very practical implementation of a concept. It seems that we are interested mainly in this, how to design the tests in practice, how to ensure that they are scientifically valid. And in most cases, this became also clear from presentations and discussions, it will be the conventional counterpart, which is the most appropriate comparator, but not in all cases. And this may even change. We are in an evolving field. We already have now cases, and the future will bring more, where this may not be possible. And I think we very rightly and intensively discussed also what is the acceptable, the scientifically justifiable approach when the conventional counterpart is not available, in order to ensure that we do a valid risk assessment. It became also clear to me during our discussion that the things are interlinked, but differentiation is also necessary in order to address the issues also in a structured approach. And I see, according to our discussion, 3 levels of differentiation. There are these general issues of GMO risk assessment. They have their values and statistical significance. What does that mean? Biological relevance, field trial design. This is one level where we need differentiation. This is not the detail of the specific issue, such as the choice of the appropriate comparator. It is linked with it, but we cannot solve how to deal with those general issues in a specific guidance document on the selection of the appropriate comparators. And another level of differentiation, which I sensed quite a lot during our discussion, is molecular characterization, food and feed, and environmental risk assessment. It became clear also during the discussion on the selection of the appropriate comparator that these issues may have to be dealt with differently. But it became also clear to me that these interlinkages between these different levels are very important and very necessary. In fact, it seems even to me that some kind of matrix or some kind of more clarification on how the various documents, regulations, guidances which we have available relate to each other. How does the guidance document on appropriate comparators, once it is finalized in the future, how does it link to the other guidances and draft guidances available? These linkages, they are very important. We have to work with a package. Finally, I think it became also clear in our discussion that we probably need also an open and justifiable process on how to deal with broader and even normative issues which are of relevance to the GMO risk assessment, to the topic of our discussion. I'm mentioning here the issue of protection goals, for example. Limits of concern. We had a very technical discussion on that during the afternoon, for example. Biological relevance. Baseline for comparison. The need for data resulting from monitoring. Who is responsible for that, for example? Or how do we define these terms and concepts? It seemed to me that we had some kind of agreement that this is not only a scientific debate which is necessary for that, and we may need different but open and justifiable processes to deal with that, with a specific role EFSA may have in that, but maybe also with some other institutions which have to come in here. I think this was really a remarkable exercise, and I think the benefit of all this is that this workshop really came in time at a stage when the draft guidance document on appropriate comparators, on the selection of comparators, is not yet finalized, but your comments which you made during the day, the presentations, the comments received, they are very valuable. They can and will be taken into account when the working group on the comparators, when the GMO panel is revising its guidance and working towards a finalization. So these are a few thoughts which I have in my head when reflecting the day. Most likely not complete, but maybe a few thought starters or issues of consideration for you for a subsequent process. I think somebody said that we are not at the end, but at the beginning of a process. I think we are within the process. The issues are evolving. For me, it was really a privilege and a pleasure to moderate this activity. I would like to thank all of you very much, the speakers, the audience. I would like to thank EFSA for giving me the opportunity to moderate this workshop. I think it was a very good exercise. Thank you very much, and I would like to hand over to Rita again.
So thank you, Helmut, for nicely wrapping up the whole discussion of today. I think you got the main points very clearly for us. The follow-up. So it's time to conclude this interesting day. I want to thank also from EFSA all the speakers and commentators, all the audience about your active contribution and discussions, critical comments, difficult questions, and really having debated and clarified your written comments in 2024 and also bringing up new issues here. So I think that was very useful for us. And all those comments are taken in due course and taken carefully for the working group of the panel and at the end for the panel when it adopts its final opinion on GM plans and comparators. So thank you for all for your contribution. It was very, very important for us. And I think also we were touching a bit about the regulatory frameworks which are underpinning all the work EFSA does, and I think that's important that we are engaging also later on in that kind of debate. But today we discussed science, and that was I'm very grateful for that one because we needed your input for this final step of this guidance of comparator selection. So how we follow up this one? All your comments will be included in a report for this public consultation and will be incorporated there. We also have published today, by your request, the PowerPoint presentations of all the speakers, and we are going to make a short summary and the webcast will be available on that site within 1 week. So everybody can follow that in the next 2 years, actually. And we have also filed a satisfaction survey because it's not the only one we keep. So please fill in both for how we organize, but also about content. That helps us to make it better and improve our systems and learn about the experience today. So please take some time and keep it registration desk when you go outside. So I would also thank also interpreters. Thank you very much for your support. It was very good and excellent. And all those who helped organize this event in EFSA, in the GM unit, in communication, and outside that one. I wish you all a nice evening, both those who were joining us via webcasting and those who are in the room, and a safe trip back home. Thank you very much.